This conference will now be recorded.
Myself, Jessica Reeves, Conference Secretariat of Coalesce Research Group LLC, very delighted to welcome you all. During this pandemic, everyone understands the importance and important role of the pharmaceutics industry and also the research provided. And it gives me immense pleasure to having you meet you all virtually at the webinar on pharmaceutics and drug delivery organized by Coalesce Research Group. Coalesce Research Group is an USA-based emerging open access publisher and international conference organizer which owed to serve the scientific community by serving the high quality, genuine research work from eminent scientists, professors, scientific research communities in all fields of medicine, clinical, life science, pharmaceutics, engineering, and technology to the research scholars, students, libraries, educational institutions, research centers, and industries with the aim of enhance your research in the respective fields. Recently, we have also started our own platform for open access peer reviewed scholarly literature where all the content is freely available without any charge to the reader. We aim to share the new recent advances through scientific publications, which will help in minimizing the borders and contributing the use of research work of eminent authors, scientists, researchers, research scholars, students, etc., across a broad spectrum of clinical, medical, pharma, and engineering scientific disciplines. And it also includes its subspecialties to promote the ideas which will guide the development and facilitate the use of scientific information and ensure its quality, and also will be connecting links between the researchers and scientific community. We are committed in ensuring that our journals are disseminated worldwide so that the researchers are able to access our content and the research content wherever and whenever they need it. We would be very glad if any of our speakers or delegates participating in today's webinar will be interested to publish an inaugural issue in any of our journals of your personal interest. So yeah, with all we'll also send you an email with all the details of the journals after the commencement of the webinar. And finally, we thank each and every one of you, the participants of this conference or the webinar. We hope you all enjoy the conference and your participation will contribute the new collaborations and research initiations. So yeah, we welcome you one and all once again. So yeah, I know virtually conducting the event virtually is really difficult, and also it's not possible for us to interact one with each and every person. So I guess it'll be better before we start our presentations. So you can just give a short introduction about yourself, so every other participants will know about you. So that'll be an easy way of communicating right now. So yeah, we have our first presentation from Dr. Stuart Cantor from Groovy Waves, USA, about the cannabis or hemp and terpenes in food or beverage, cosmetic and pharmaceutical products. So yeah, Dr. Stewart, you can actually start sharing your screen. You can just click on the screen
Okay,
button.
so I'm gonna do that, and then
Yeah. Hmm.
how do I need to make
Yeah.
this minimum. What is?
Oh yeah. In the above, you will have an option of view everyone. So you can just select it
Okay,
as hide everyone. Yeah, it'll be clearly visible to you.
okay,
Yeah.
okay. So I guess you tell me when I should start.
Yeah, actually we can start a presentation now.
Okay, okay.
Yeah.
Well, good morning, everybody. I'm Dr. Stuart Cantor. I'll give you a brief intro to me. I have a PhD in pharmaceutical sciences. I worked in pharma for 10 years. Last company I worked for went bankrupt, and the way I got into cannabis was 3 months later, I was driving home one day, and a Colorado CEO called me out of the blue and said, "Can I develop cannabis products?" And I said, "Sure, I've done plenty of other things." So I started working with him in my kitchen, and Then I got another 9 companies, and the Maryland company, Ruby Waves, was one of the companies that I consulted for, and then they made me chief scientist in December, and I moved back to Maryland. So the cannabis hemp, the plant is really great. There's a lot of chemistry involved, and so I'm going to share some of the things I've done and basics about cannabis for those who may not be too familiar with it. So here's a nice overall chart showing at least 10 cannabinoids. There's thousands of terpenes. And our lab tests cannabis. We do full testing. So basically that involves potency, heavy metals, pesticides, residual solvents, mycotoxins, bacteria, everything. So it assures the public that our cannabis extracts are the highest quality. This slide shows, similar to dietary supplements, shows how some of the public feels about CBD or even THC, that they want the ability to choose what they put into their body. And there has been research to show, especially with opioids that are addictive, that taking cannabis can also help with pain and help wean people off any opioid addiction. It was really interesting for me to learn the rich history of cannabis going back thousands of years. I mean, it originated in, it's been used since ancient times in India and China. It was even part of the USP until 1942, so doctors were prescribing it. So we can fast forward to 2018, FDA approved a purified form of CBD for use in epilepsy and seizures. However, there were several other synthetic cannabinoids approved by FDA in the mid-80s for cancer and cancer-related pain and nausea. So here's an outline. I'm going to discuss the basic botany, chemistry, PK, the ECS system, regulatory. In the US and also in other countries, the regulatory and banking is still in a state of constant flux. I'll discuss that briefly, and the range of products that can that cannabis can be in. You know, either edibles or personal care cosmetic products. So sometimes people still get confused about cannabis, hemp, marijuana. So marijuana is the cannabis plant, but it's sexually bred to contain a high level of THC, which is a psychoactive compound. Other strains are selectively bred to be predominant more in CBD. And sometimes, obviously, the plant can have both, but the extracts can be a predominance of CBD with some levels of THC that are still acceptable federally. So for foods, we can have edibles, we can have tinctures. Typically, tinctures is we can have the MCT oil in dropper bottles that has the cannabis in it to put under your tongue. We can have personal care products, a wide range, so we can have skin creams, bombs or gel. We can also have pharmaceutical supplements, capsules, and transdermal patches. So basic botany, there's 3 main strains of cannabis. Sativa, which is a source of hemp, so that is hemp that is compliant with the 2018 Farm Bill in the US, so it's less than 0.3% THC by weight. In Europe, the level is different. It has to be THC percent. Hemp seed oil, which is a good source of omegas, is not psychoactive at all. The cannabis itself, the CBD and THC come from the trichomes in the plant. We can have indica, and some people going into dispensaries will specify, you know, they want a high sativa, high indica, they want a combination of both, depending upon what type of condition they want to treat. We have ruderalis, which is a low THC species of cannabis. And again, the predominance of CBD and THC has to do with how the plants are bred. You have one or both as predominant compounds. So with sativa versus indica, the plants are actually different. Sativa, the plant is tall and stature with narrow leaves. Indica it's shorter with broader leaves. So you'll be able to tell the difference by that as well as the shape of the leaf in the plants. So we have some different terminologies. Full spectrum CBD is CBD along with the whole plant. So that includes the terpenes, fatty acids, and again, to be compliant with federal regulations, it should contain less than 0.3% THC in the US. We have broad spectrum CBD. That is similar, although the THC content is essentially zero or not detectable. However, when we extract out the THC, typically a bunch of the terpenes also go away because they have lower boiling points. Industrial hemp is the cannabis sativa with less than 0.3% THC. It can be either grown as a fiber or as a seed crop. Isolate is a high purity 99% extract of THC or CBD as a powder. Currently, there's no standard of identity for full or broad spectrum, but I'm part of the ASTM committee for cannabis, so we're discussing terminology for hemp seed oil and all aspects of cannabis. So there are over 100 different types of cannabinoids with 10 major cannabinoids. There's a bunch of minor ones. We have the synthetic THC dronabinol, which was approved by FDA in the mid-80s. We have Delta-9 and Delta-8. Delta-8 is less potent than Delta-9, so typically if we talk about the level of THC in a product, it's typically the Delta-9 we're talking about. Both the CBD and THC have acid forms, which must be decarboxylated because only the THC and CBD non-acid forms are bioactive in the body. And we have some minor cannabinoids, CBN, CBG. In January, there's a publication came out talking about THCP and CBDP, which some scientists were saying they could be up to 20 times more potent than the CBD or THC, so I thought that was very interesting. So if we look at the basic chemistry of the molecules, they are similar. The differences are highlighted in the red circles here, and they both have the same hydrocarbon tail at the end. However, it's the THC that's psychoactive, Schedule 1, not CBD. If you're new to THC, you should start out slow until you know how your body reacts. You should take it with food. Start out with 5 milligrams or less, depending upon your age and weight, and see how you react with it. If you take too much THC, you won't always get the giggles or munchies or feeling good, because THC is rather powerful, as I've discovered. So it can also cause nausea, dizziness, and low blood pressure. So that's why I say you should take it with caution so you know how you're going to react with it. The CBD is not psychoactive. It treats pain, insomnia. That's the purified form is the Epidiolex, approved by FDA a year ago, 2 years ago. And the CBD can treat anxiety, inflammation, pain, can help with sleep, and they both can work synergistically together. So the CBD can help modulate the effects of this THC. A little bit about pharmacokinetics. The cannabinoids and terpenes are highly lipophilic, but they have a low oral bioavailability. If you were to smoke cannabis, you find that within 10 minutes you're going to start to feel the effects. But edibles are different because the oral absorption can be slow and erratic. Peak levels can be reached typically between 1 to 2 hours, so you're going to have a delayed effect. Also, cannabinoids have extensive first-pass metabolism. However, if you put a patch on your skin or rub it into your skin, that can bypass the hepatic route. It's interesting that the oral half-life depends upon the frequency of use. So for CBD, it's about 1 to 2 days. However, for THC, if you're an infrequent user, half-life is about 1.3 days. But if you're a heavy user, it can stay in your body for 5 to 13 days, even longer. With the terpenes, there's thousands of terpenes, about over 200 terpenes in the plant. They're also lipophilic. They can be extracted via different ways. The terpene profile is what's unique to strains. I don't know, I think about 5 years ago, companies had started adding back some of the terpenes to their food products to give them like a niche in the market. So as far as the whole plant is concerned, the terpenes can work together with the cannabinoids to give the desired effect in the body. And different strains will have different levels of the different terpenes. So here's the basic structure of some of the terpenes, and some of you are already familiar with some of the terpenes, like pinene and pine sol. That's pretty characteristic. Myrcene is the most abundant. It's also found in mango. These all have different boiling points and vaporization temperatures, and herbalists have said that they have different types of potential medicinal values for different treatments. So limonene from citrus, beta-carotene is in pepper, humulene is in hops. So say, for example, humulene is thought to have some anti-inflammatory properties, whereas some of the other ones, alpha-pinene is for pain, as well as beta-carotene. I published an article on cannabis products in 2019 about using terpenes in edibles and beverages. It's on my LinkedIn profile if you want to look at it. Now, the endocannabinoid system, or ECS, was discovered in, I think, the late 60s. So it's been known for a long time. Even CBD and THC were discovered in 64. So the knowledge has been around for a while, but it hasn't been obviously widely publicized about some of the science for quite some time, only until the last couple of years when the explosion of interest in cannabis has been global. Cannabinoids can act as neurotransmitters. We know there's different types of receptors depending on where they are in the body. We have CB1 and CB2. THC binds to both receptors. However, CBD has no particular binding affinity. So there's been a lot of research on the ECS system as it's present in both humans and in animals. This is a schematic showing the ECS system distributed throughout the body. You can see it's quite extensive. Now, the entourage effect was coined in the late 90s. It describes the interactive synergy between the cannabinoids and the fact that CBD modulates the effect of THC in the body. The cannabinoids also synergistically work together with the terpenes and whole plant medicine. So this is something I believe. So basically, like, you know, when you take it's better if you take a extract that has the terpenes in it, or even if they're added back, but the whole plant works together to give you the therapeutic effects. It's not necessarily just an isolate. The whole plant is preferential rather than isolated chemicals. So if we look, the market size and valuation of the cannabis market is constantly changing, growing throughout the world. It is big. In 2017, it was about 10 billion. 2019, it was about 17 billion. I'm not sure what it is now, but the interest in cannabis has been growing around the world. All the major players are eyeing aspects of cannabis, either pharma or even food companies, how they can incorporate it into their business. And here you can see a comparison. Look at the legal marijuana sales at 3.4 billion. This was in 2015, actually. You can see even that outpaces things like Dasani, which is 1 billion, Oreo, which is 711 million, and other types of foods in the U.S. This is the legal state of cannabis as of last year. I'm not sure if some of this is changed a little bit. So we're talking about the legal status of cannabis. We have recreational use, medical use. We have to have a card. Different states have different requirements. There's a bunch of legal stuff around the cannabis plant. So FDA has actually had several approvals of cannabis-derived products. The first 3 were approved in the mid-80s and also 2016 for cancer, chemo, nausea, pain. These are all synthetic cannabinoids. Epidiolex is a purified form of CBD. The regulatory status is always changing. Different states have different requirements. So the symbols up at the top, the states require that if you have any edible or tablet or any type of food product or pharma, that each dosage unit should be labeled with a THC symbol so that the public knows that it is THC in there and that it can cause psychoactive effects. Again, FDA never said that cannabis is allowed in food and supplement products. They never said you can call it a supplement. According to them, they said it's not eligible to be grass. Perhaps grass affirmed, but FDA has not come out with their final stance on their opinion of cannabis federally at this time. However, perhaps in another 1 to 2 years, they will have some kind of path forward and guidance via Congress. Again, a lot of companies will put this disclaimer on their products, typical of what is on products for dietary supplements, that the statements have not been evaluated by the FDA, and you may not modify the wording of the disclaimer. Again, the THC and CBD are not regulated under Dietary Supplement Health Education Act, and if you are labeling, making claims on your product, you need to be careful. You don't want to be making drug-like claims. FDA has not really has gone after companies for making gross misstatements, such as cannabis can cure COVID or different things. You know, if they do product to a disease, they're most likely going to get a warning letter. So you want to avoid that. The things you can say about cannabis are can help support a healthy mood, help support a healthy sleep pattern, things like that, that support the body, but not make drug-like claims. Typical government warnings about cannabis is the fact that the intoxicating effects of cannabis may be delayed for up to 2 hours, especially true if you're taking it edible. There is a delayed reaction. Nutritional facts panels are mandatory on cannabis edible products, so the public needs to know what they're taking. It is a requirement that the milligrams of CBD and THC per piece are disclosed, as well as the total amount of those in the product itself, clearly labeled on the package, along with the expiration date, etc. Cannabinoids and terpenes are highly lipophilic molecules. You can see on the right, this is a picture of canna butter. So it's cannabis infused into an oil or butter for baking. And again, it's only the decarboxylated forms of CBD and THC that are bioavailable and active in the body, not the acid forms. For emulsions, we can use synthetic tweens like polysorbate 20 or 80. With different HLB values. I'll discuss that in a minute. The natural alternative is Coalesce extract. However, it does taste bad, so I don't particularly like using it because for an oral product, however, it can be perhaps fine for a topical product. Again, with oral products, some terpenes have better flavors than others. Cannabis extracts can have some off notes and tastes, so it's important to control the quality of the extract that you're going to use if it's going to be a food product that someone's going to put in their mouth versus something on their skin. And you want to check on the C of A's so you know how much you have in the product. This is an HLB scale. So HLB stands for hydrophilic lipophilic balance. It goes from 0 to about 18. And the midpoint is about 9, so basically above 9, we're talking about oil and water emulsion, so the water is the bulk, the higher phase, and below 9, we're talking about water and oil emulsions. So this is typically more products that are more hydrophobic, like Antiphon has an HLB of about 2 to 3, whereas detergents have an HLB of 13 to 15. Polysorbate has an HLB of like 15.5, tween 20, polysorbate 20 has an HLB of like close to 17. So as we go higher on the HLB scale, the wettability and solubility greatly improves. So with an HLB, we want to understand the level of the percentage of oils in the oil phase, what total contribution we have in the oil phase of the total HLB from the oils, and then what's surfactant or emulsifier we're going to select. The HLB of the oil and the surfactant should be within 1 unit of each other. We should choose a combination of surfactants, both a higher and a lower HLB for a more stable emulsion. So the two ways that we can put energy in the system to make a oil and water emulsion or even nanoemulsion, microemulsion, is either high pressure homogenization or sonication. I use both in my kitchen. I was surprised to find that the high pressure homogenization for the emulsion that I had really did not work. It was the ultrasonication that gave me the best results to make a nanoemulsion. On the left-hand side here, we have the horns, which are used to generate the sound waves for ultrasonication, and the schematic is at the bottom here. You can see the sound waves cause cavitation of the oil droplets to shatter and break apart to make them smaller so that it can give a more stable emulsion. On the right, we have a rotor stator, which is the principle of a high-pressure homogenizer. This is an example on the left of the high-pressure homogenizer I used in my kitchen. On the left is a R&D model. On the right is a large industrial model. If we're talking about using cannabis extracts, again, there's a process that we go from R&D to commercial scale. So on the left is about 500-gram vessel, glass round flask versus 50-liter flasks on the right. The Colorado CEO had a 50-liter rotovap on the right in his factory. It was nice to be able to see that. Okay, so as we talk about HLB, typically, if we have an oil and water emulsion before we put energy into the system, it's typically going to be opaque dispersion. So HLB would be typically within the range of 6 to 8. To be stable, 8 to 10. So as we put energy into the system, we're going to see that the cloudy dispersion is going to become more translucent. And then once we see that, we're going to know that the particle size is typically in the nano range, even below 100 nanometers. On the right-hand side, we have the oil and water. Oil has a lower density than water, so it floats on top. We can see here the oil droplets start to align here, where the hydrophilic heads are pointed into the bulk water phase, and the hydrophobic tails are pointed toward the oil phase. And here we have the formation of micelles, where the oil is protected inside, and the hydro polar heads are toward the bulk water phase. So we have something called the critical micelle concentration. Once we have a sufficient level of surfactant, we can have a tendency to form the micelles, which are stable. They're about 2 to 20 nanometers, so they're very small, and that's how the solution will appear to our eyes as being more translucent. Here's some theory about the CMC. So basically, we have the air-water interface. We start to have the alignment of the polar heads and the bulk water, and the hydrophobic tails are going to go to the oil phase. And as more energy is put into the system, if we have sufficient quantity of surfactant, we'll have the preferential formation of the micelles, which helps stabilize the emulsion. So here's an example of what I'm talking about. So ultrasonication, or high pressure. I started out with a opaque dispersion, and as I put more energy in the system through multiple passes, you can see number 5. You can read newsprint through it. So I knew that I must have a nano emulsion at that time. So I had the particle size checked, and it was about 56 nanometers. So it was nice to be able to make that in my kitchen, and it was the ultrasonication that enabled me to do that. For the same formulation, the high pressure was not as effective. Okay, so a little bit about food science. So we have different types of emulsions we can make, and the instability can be introduced through gravitational separation, coalescence, flocculation. Osmolar ripening is where small oil droplets dissolve and deposit on the larger particles. For an emulsion, a coarse emulsion, a conventional emulsion, the oil droplet size can be greater than 1 micron. It'll appear cloudy because the particle size of oil is rather large. The interfacial tension is high. Microemulsion and nanoemulsion, although a lot of literature can consider the oil droplet size to be the same, 20 to 200 nanometers. Typically, a microemulsion in a food product is, we think of that as like being less than 0.5 micron in size. These can form spontaneously. And the difference is the microemulsions are thermodynamically stable. However, the nanoemulsions typically require higher surfactant levels. You have to put energy into the system to get the particle size down that low. And they're kinetically stable. So if the formulation isn't right, or the manufacturing conditions aren't right, they can slowly degrade over time and cause some instability or separation. So we'll talk about some cosmetic science. So in order for cannabis creams to penetrate through the stratum corneum, typically they'll have a penetration enhancer. The pH of your skin is a little bit on the acidic side, so it tends to be pH about 5.5. Now we have all different types of topical products. We can have creams, gels, ointments, and balms. And we have a variety of different emulsifiers to choose from. We have amphoteric emulsifiers like lecithin, which have both positive and negative charges. We can have amphophilic emulsifiers, which are both hydrophilic and hydrophobic parts. We can have non-ionic, cationic, or anionic emulsifiers. Again, the polysorbates are between 20 and 80 are not considered natural. The Coalesce is. However, it may not be effective, as effective as the synthetic emulsifiers. And we have other ones. We have cetearyl alcohol and ceteris 20, which is a combination of fatty alcohol and an emulsifier in one. It has a rather high HLB, which is similar to polysorbate 80. We have viscosifiers and polymers in our emulsion, starch, xanthan gum, carbomer. We have emollients, which adds smoothness, a smooth feel to shea, cocoa butter, lanolin, petrolatum. We have skin penetrants, which help the oils go through the stratum corneum into the skin. IPP, isopropyl palmitate, isopropyl myristate. And we have preservatives, which are critical for oil and water creams or products, because if we're going to keep on putting our fingers into the cream, it must have preservative. A skin balm or salve is non-aqueous, so it's not required that preservative be in there. Across from the supplier of squalane, they measured some CBD penetration through the skin, and they found that the squalane added had a 14 to 40 time improvement in the delivery of CBD. So that was very promising, even at 1% of squalane. Sorry, yeah. For a cannabis product, it's important for a manufacturer to know your supply chain. So you want to get a C of A from whoever you get your extract from. You'd be good to audit your suppliers. And again, you want your cannabis extract tested for potency, pesticides, heavy metals, mycotoxins, bacteria. residual solvents, everything, so you know that it's within compliance. And the cannabis plant does tend to absorb things from the soil. That's why it's important to test for all those things so you have assurance that your cannabis extract is the highest quality you can have. On the left is a Mood 33. It's a sparkling beverage. I covered that in my cannabis terpene article. So basically, this manufacturer is adding back specific terpenes for different effects. The bottles are labeled joy, passion, and calm. So different terpenes are added back to, as I said, the terpenes can have different beneficial effects on the body. People have been adding cannabis to wine and beer, and also tea on the right. Here's some more products that are out. Gummies are popular, sparkling waters, baked products, everything really. Kiva chocolates, I tried those when I was in San Diego. Those are good. Chewing gum. These are pillow type of gum with a crispy coating on the outside, like a Clips gum. I never tried them, but I've tried other cannabis gums. And again, from trade shows, we can get tinctures in a dropper bottle, soft gels. They were handing out syringes of the natural patches, creams, anything really, any food or edible product that you can use, you can put cannabis in. I'll discuss briefly about tablets and capsules. My background when I got my PhD was in industrial pharmacy, so formulation and manufacturing of tablets, capsules, and coatings. So tablet and capsule formulations are somewhat similar. They have similar excipients. For the API, you can have low dose or high dose. An example of a low dose drug is like levothyroxine synthroid. So that can be like 25 microgram to 30 to 300 microgram. And with low dose drugs, it's important to have your manufacturing done a certain way to make sure you have content uniformity, better improve content uniformity in your final product. So here we have your fillers, MCC, silicified MCC, lactose dical. If you're doing granulation, it can be wet or dry. You have different polymers and binders for that. Disintegrants, lubricants, glidants for improved flow, silicon dioxide or talc. So if you have a low dose API, such as some microgram dose, you want to use geometric dilution to blend that initially with half of your excipients and keep on going until you blend all of your excipients together to ensure improved content uniformity in your final product. You want to sieve your magnesium stearate through a 30 to also enable it to be dispersed properly throughout the powder bed. The next stearate level can vary between 0.1 to 2%. So there's a wide range there. And you should do an optimization study to see, because it's advised that you avoid overblending, because that can negatively affect the plug formation in capsules and also dissolution. So it does have an effect. It does have a hydrophobic effect on the product, although it is required for processability with the machines to lower some of the friction. You can use dical, however, it's rather abrasive. You should keep it less than 15% in the formulation. You want to do a product size analysis. If you do granular lines in the oversize, and when you do your final blend, you want to check for segregation to make sure that that's not going to be an issue with the content uniformity and weight uniformity of your tablet or capsule products. This is some work I did at FDA, and this is chemical imaging. So in here, I was blending coated drug beads with different levels of placebo wax beads and forming tablets. So you can see here I was supposed to be going for a 20% drug bead in the tablet. So you can see segregation was occurring as the tablet press was running. I was pulling out tablets from the beginning, middle, and end of the run. So here you see the actual average is about 29%, although I was supposed to have the blend as 20%. Middle was 22%, and the last was 19%. So you can see that it's important to control the level of segregation, blend segregation while the tablet press is running. You should know that beforehand before you go to commercialize it. For tablets, there's a bunch of physical chemical parameters you want to test in the lab to make sure that your performance is okay. And also USP monographs will also have some information about this in the USP. So you test for hardness, friability, disintegration. If you have an ODT, that would typically be less than 30 seconds dissolved in the mouth. Dissolution, content uniformity, weight uniformity, and stability testing, both ambient and accelerated. There's different types of tablet defects we can have. It's thought to be primarily due to air entrapment if we're talking about capping and lamination. So capping is when the top of the tablet is sheared off. Lamination is when there's instability throughout the tablet causing weaknesses. You don't want either of those. You can also have sticking and picking. So, that's why it's important to do a lot of evaluation when you're doing the R&D scale on the tablet press, and then when you're going to scale up, you're going to be checking to make sure that your tablet products are okay. For capsules, the fill depends upon the powder density. We have a variety of different capsules for both human and animal that have different volumes. And for tablet and capsule products, you'd be surprised that you're not going to be able to fit everything into there that you think based on the flowability, the level of active. So you may decide that you want to split the dosage form of your actives up into 2, 3, 4, even 5 dosages in order to get for certain high dose products. You may have to do that. In conclusion, you want to start slowly on edibles, especially with something that's psychoactive like THC, until you know how your body reacts. So start with 5 milligrams or less, and remember that there is a delayed effect of 30 minutes to about an hour and a half before you're going to start seeing any effects in your body. Microdosing is a hot trend. The effects will vary. It's your choice whether you want to use a whole plant extract or an isolate. And again, whether you want sativa, indica, or a combination of both. When I tried to find some contract manufacturers, 2 years ago, some were not willing to work even with CBD. Some of that has changed. Again, the regulations, the legal status, the banking are continuing to evolve around the world. Even some of the big food companies have been eyeing cannabis because of the high profit it can bring to their products. Coca-Cola, Mondelez, Kraft, and Nabisco. Who knows what's going to happen in the future when they come out with their products, but we'll see. Carl's Jr. developed a CBD-infused burger on April 20th or 4/20 last year. So cannabis is big business in both food and pharma. So I've written 2 articles on cannabis, one for the terpenes on my LinkedIn site, and also for Techno Agroscience. It was handed out at VitaFoods Europe. It was talking about, I interviewed thought leaders and wrote my opinion about what's going on with cannabis regs and what's going on in Europe. Pharmaceuticals is the company of the Colorado CEO. He makes an encapsulated cannabis powder for food and personal care products. Steve Rowley has his own lab. He does spray drying, freeze drying, and microencapsulation. I had spoken at the CBD Expo last year in Orlando, and I also spoke in Orlando again at a food convention, IFT. So I've developed a variety of different products in my career: tablets, capsules, coatings, which were launched by Bristol Myers in 2003. Nutritional bar, different types of powdered beverages. And I'd like to thank Coalesce Research for allowing me to speak to everybody, and thank you very much.
Thanks a lot, Dr. Stewart. Thank you for your presentation. It was really informative to know about the cannabis and food products. Yeah, thanks a lot. So, do we have any questions for Dr. Cantor? If any of you want to speak out, you can just click on the mic button present on the bottom. So you can just speak out if you're having any queries. So yeah, I hope there are no queries, Dr. Stewart. So yeah, maybe you might face some issue asking directly. So you can also we'll be sharing you all your email IDs to your email so that you can just contact them in any case if you're having any doubts or for any collaborations or anything. So thanks a lot, Dr. Stewart, Cantor, for your presentation. It was really impressive.
No, thank you.
So yeah, moving on to our next presentation. Yeah, we have Dr. Diana Harahap from University of Indonesia about the volumetric absorptive microsampling as a sampling alternative in clinical trials and therapeutic drug monitoring during the coronavirus disease pandemic. That's a review. So yeah, Dr. Yardiana, you can actually, can you hear me? Like, please unmute your button.
Yeah. Good morning.
Good morning, Doctor. Doctor, you can just click on the screen button present at the bottom.
Button
button.
press.
Yeah, screen. You can see the word screen
Oh,
on
screen.
Yeah.
Yeah.
Please do click on that. So
Screen.
you'll be able to see your screen, like whatever screen you would like to share with us, you can just click on that. Were you able to do that?
Okay.
You could just click on, yeah, it's visible, it's perfect.
Okay. Can you see my slide?
Yeah, we can see your slide. Yeah, that's perfect. It's visible.
Okay. Thank you.
So
yeah. It is midnight in Indonesia, so I'm sleeping.
I completely understand. Really sorry for the inconvenience, but yeah, we have a time difference right now, and we'll make sure we'll work on that for the next time for sure. And we're sorry for the inconvenience.
Okay. Okay, so
Please start the presentation. Thank you.
Yeah, I'll begin. So, good morning, everyone. Today, I'd like to present our review of the volumetric absorptive microsampling as a sampling alternative in clinical trials and therapeutic drug monitoring during the coronavirus disease pandemic. This is an article review. I'm Yahdiana Harahap from Faculty of Pharmacy, Universitas Indonesia, Depok, Indonesia. So our lab is a bioanalysis laboratory. So this review, I met with Rasmina Dipta Sadia. This is my students. So I'll continue. This is the outline of my talk. And for the introduction, as we know that the coronavirus began on December 2019 in Wuhan, China. The symptoms is fever and dyspnea, and is caused by virus, this severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. And there are many cases of pneumonia. Assumptions comes from seafood and like in animal market. And 2020, it stated that the virus is we said COVID-19 because it's happened in 2019. And the disease transmission from human to human, if the droplet in the 1 meter, so social distancing it's needed because shorter than 1 meter, the droplet without mask will touch another human and can cause the SARS-CoV-2. And according to data from Johns Hopkins University, USA, on 10 September 2020, from 188 countries or regions, it is around 27 million, around 27 millions people affected by this virus, and costed about 200,000, around 200,000, and recovered by 18 million people. While in Indonesia until right now, about 203,000 that affected by this virus, and died about 8,000, and recovered about 145 people. The symptoms from moderate to severe respiratory distress, fever, and also followed by cough and nasal congestion, and also fatigue. And there's also the severe symptoms such as pneumonia, and this pneumonia can cause death. And how about the prevention? The prevention is washing hands with soap or alcohol-based hand sanitizer, and also maintain the social distancing around 1 until 2 meters, and also wear a mask. And the medicine and vaccine is in clinical trials until right now. And the WHO in 11 March 2020 stated that it is the global pandemic, and need to quarantine and work from home, and closing the crowded places, travel restrictions, and also hamper clinical trial and TDM protocols. And the protocol has deviations from the normal protocol. And then we continue to the biosampling technique. For accurate drug quantification in matrix, an important aspect of clinical trials of drug development and TDM, for the pharmacokinetic studies, pharmacodynamics, and toxicokinetics, there are many kinds of usable matrices such as blood, CSF, breast milk, saliva, urine, and hair. But until now, the golden standard is the golden standard for matrices are plasma and serum. But this matrix has some disadvantages, such as invasive, need medical assistance, storage conditions must be appropriate, and need large sample volume. So we need an innovation, such as dried blood spot or volumetric absorptive microsampling. So what is dried blood spot? Dried blood spot, a blood microsampling technique that involves collecting blood on a small filter paper. The advantages: less invasive, small sample volume, and can be stored and distributed at room temperature. But there are some disadvantages. There is an effect of hematocrit, This hematocrit plays a role in the determination of viscosity, and can affect the distribution of blood samples on the DBS paper. So it can trigger the bias in analysis. And what is volumetric absorptive microsampling of FAMS? It is a blood microsampling technique that involves a precise volume absorption of blood using hydrophilic tips. The advantages, less invasive, same with the DBS, less taking volume, and easy in handling. Accurate sample volume collection reduces the effect of hematocrit, no need for medical personnel. The sampling can be done at home, but this also has the disadvantages such as expensive sampling can only be processed once. So because this is due to the COVID-19 pandemic, so it has the advantages because we can do the sampling at home and it can slow down the chain of transmission of COVID-19. And how about the analysis? We can analyze it with liquid chromatographic tandem mass spectrometry or LC-MS that has this high sensitivity and selectivity. And how about the challenges during the COVID-19 pandemic, social distancing, but this situation can inhibit the implementation of clinical trials for drug development and therapeutic drug monitoring. And FDA stated that the sponsor or researchers should consider alternative study workflows during possible disruptions. So we need the technological development and innovative approach to increase the productivity of the implementation of clinical trial protocols for drug development and therapeutic drug monitoring. So this is the innovative technology for biosampling technique. This is volumetric absorptive microsampling. This is a sampling alternative for clinical and bioanalytical aspects. So we want to see about the clinical aspect. First, the clinical consideration for implementation of volumetric absorptive microsampling to clinical trials and therapeutic monitoring during the COVID-19 pandemic. If we see the advantages of FAMS in clinical trials, minimally invasive, small sampling volume, sampling only from the tip of the finger or heel, and can be done at home. And then this is the suitable approach for vulnerable populations such as pediatrics, geriatrics, patients with acute episodic disease, example, epileptic patients. And also the patient will be comfortable for this microsampling because the sampling is faster and easier without the help of medical personnel and small sample volume around 10 until 30 microliter of blood. Because of that, can save sample storage and shipping costs. Because the samples can be stored at room temperature, delivery in dry samples, patient can collect their own samples at home, save travel cost. But the sampling tools are expensive, it is relative, and can only be used for one sample. And also needs sampling handling and preparation requires special expertise to get maximum results. So for this method, the sampling tools can only be used for one sample, so we do the sampling more than once. So actually, there are beside the DBS and FAMS, this is the standard, the golden standard for biosampling is venipuncture. So venipuncture, the blood collected from the venous, and if we compare the three of these biosampling technique, we can see here that in venipuncture we need the centrifugation for analyte separation. if we want to take the plasma or serum. And then the storage and delivery, we need to keep the plasma or blood from venipuncture in freezer, while in DBS and farms, we just need to store it in room temperature in dry samples. And for the sample preparation for venipuncture, if you want to get the plasma, we should add the anticoagulant to get plasma. While in DBS, sub-punching method, cutting the DBS to consistent diameter from the paper. And for farms, just separate the tips from the handle. And for the sampling, for venipuncture, invasive, and large sample volume until 5 milliliter. While in DBS, minimally invasive because we can collect the blood from finger, so finger prick, and small sample volume around 10 until 30 microliter or 50 microliter. And farms, minimally invasive is same with DBS and small sample volume 10 until 30 microliter. Second, the clinical consideration for implementation of FAMS to clinical trials and TDM for COVID-19. So in this pandemic, there are many drug development for COVID-19. So should involve high-risk patients such as pediatrics, geriatrics, patients with comorbidities such as cardiovascular, respiratory disease, chronic kidney disease. So to provide minimal treatment as possible, we can use FAMS for the sampling technique because increased risk. If we see this slide, for the drugs in clinical trials, until right now is remdesivir and chloroquine. Actually, chloroquine for anti-malaria, but now it is also proposed for the COVID-19. And these drugs must pass the clinical trials. So it needs the drug safety, biopharmacokinetic profile, and drug effectivity. And the TDM must be done because chloroquine can cause heart problems if the drug concentration in plasma 65 until 263 nanogram per ml. If over this concentration, it can cause heart problems. And from the many literatures said until 700 nanogram per ml, it can cause heavy heart problems. In the implementation of clinical trials for drug development and TDM, consideration of cost effectiveness and ease of sample handling is needed because this test can last for months. So we should try to find the cost effective. Because of that, we need farms can be used to reduce research costs because less files, less sample cups, and sample preparation tools are used. No need for anticoagulants, freezers, and centrifuges. So the NIH started a study to measure undetected cases of COVID-19 infection. So determine how many this can determine how many people have no history of COVID-19 symptoms but have antibodies to the virus. So it is already analyzed farms blood samples from 10,000 volunteers, and the study will provide an overview of the magnitude of the COVID-19 pandemic in various infected communities without knowing it. So the laboratory tests for COVID-19 such as swab tests. The swab test is unable to detect previous infection. And another is serological test. It is detect of antibodies from serum, from venipuncture sample. And then the NIH study by farms and can analyze the antibodies to the SARS-CoV-2 virus. So the use of FAMS is safe, effective, and easy in sampling. Patients can take their own sample from home. Third factors to be considered in the use of FAMS for clinical trials and TDM during the COVID-19 pandemic, factors affecting volume variation. The FAMS sampler tip must not be immersed in blood, maybe absorb excess of blood. So utilizing the media to socialize the proper and correct use of FAMS to users, and also need to do the online training forums, video tutorials, and instruction brochures. And also it is need the volunteer compliance factor. In Indonesia, the discipline of the people is low. So we should monitor the use of these farms, and also they must complete and informed consent. The patient is deemed to have complied with the research objectives, risks, and procedures. And also because there is no direct control during a pandemic, so the effort, periodic control via telephone contact or online forums, or give reminders to the volunteers. So the Session B is bioanalysis aspect. The role of the bioanalysis laboratory provides quantitative measurements of drugs in a matrix with suitable analytical method so as to produce accurate data. In microsampling studies, this requires validation of the method to ensure the sensitivity and selectivity of the analysis because fewer samples were used. So the method validation must overcome the effects of storage and delivery temperatures, sample homogeneity, and also HCT hematocrit effects and stability. The bioanalytical consideration for the implementation of FAMS in clinical trials and TDM should do the fixed volume uptake and stability of analytes and metabolites in the FAMS, and also homogeneity of sample and hematocrit effects, and carryover and cross-contamination, and also logistics. And bioanalytical consideration for implementation of FAMS in sample analysis, accuracy and precision, recovery, and also the matrix effect if you use the LC-MS-MS. First, the bioanalytical consideration for implementation of FAMS in clinical trials and TDM, we should do the fixed volume uptake. For the conventional sampling, such as venipuncture, require high accuracy to get accurate sample. Sampling and handling should be handled by trained analysts. While in DBS, the diameter of each spot is different on different papers because there is the effect of the hematocrit. And also use the sub-punch technique to get identical dots. While FAMS available in 10, 20, and 30 microliter volumes, and also must be monitored for the use of FAMS because the blood should be fixed in 10, 20, and 30 microliter of volumes. And about the stability of analytes and metabolites influenced by temperature, humidity, sunlight, storage, and shipping condition, and also chemical reaction and enzyme catalyzed degradation. In conventional sampling, can minimize the degradation by adding enzyme inhibitors, controlling sample pH, and antioxidants in the collection tube. And in macrosampling, addition of these components during extractions allows degradation when drying the sample. So this is the consideration if we want to implementation the FAMS in our study. Stability of the drug maltofosine in FAMS and DBS in This is the research by Kip et al. In 2017. FAM stable for 1 month at room temperature, while DBS is stable longer than in FAMs. It is about 5 months in an aluminum bag at room temperature. The stability different analytes in FAM sample depends on the type of analyte being studied, and the stability of analytes cannot be ascertained for a long time at room temperature. So DBS in the stability of analytes and metabolite is better than FAMs. Homogeneity of sample and hematocrit effects. Blood with different hematocrit values affect blood viscosity, and it will be varies in the size of spots scatters in the paper of the DBS, and it can trigger the bias in analysis. High hematocrit concentration will give high red blood cell count in the paper. So it can trap on the DBS paper substrate and can inhibit the analyte desorption process. And can affect the recovery of analytes. So if we see the research using human blood and mice with different hematocrites from 20 until 65% absorbed by FAMs, this is by Spooner et al. 2015. The human and mice blood with different hematocrites absorbed by FAMs were investigated in 6 different laboratories. We can see here in this figure the gravimetric determination of the volume of blood absorbed by volumetric absorptive microsample tips of various hematocrit values. We can see here in this figure that from one laboratory to others, the slope of human blood volume to hematocrit probe is close to 0. But the volume of blood collected by FAMs has little difference. If we see here, this is the dot, has little difference with the different hematocrit blood values. So if we see the percent CV, the coefficient variance, it is around 8.7%, but it still meets the coefficient variance precision requirements of not more than 15%. And how about the carryover and cross-contamination? If contamination occurs during sample collection, it can cause experimental errors and then affect the selectivity and accuracy of the analysis. And sampling technologies should be designed to eliminate or minimize the contamination. Most DBS designs are open, so the paper is open and have no special protection. Contamination may occur during the drying time because for the drying it need around 2 until 3 hours. While in farms, you can see here, this is the tools equipped with a cartridge protector. Allows drying of the sample in closed condition. It can minimize the contamination from the environment. And how about logistics? Farms can provide practical benefits. Simple sampling, samples can be sent and stored at room temperature, can collect samples by yourself at home, equipped with a collection device and a shipping container, and easier for users to use it, and sending samples to the laboratory, and cheaper because it can be done at room temperature. And second, bioanalytical consideration for implementation of FAMs in sample analysis. This is accurate and precise, and the criteria, nominal concentration, and the coefficient variance are not more than 15% for Q samples and not more than 20% for the LLQ samples. The accuracy and precision of the for the maltodextrin in FAMs and DBS by Kip et al. 2017. For DBS, the nominal concentration is not more than 15% for all samples, and 7% for the LLOQ, and the percentage of coefficient variance for precision not more than 19%. And then we compare with farms. Farms, the nominal concentration is 10.8% for all samples, and for LLOQ 7.9%. And for the precision, not more than 11.9%. So if we compare DBS to FAMS, FAMS is more accurate and also more precise. And recovery and matrix effect, if we use the LCM-SMS, we should investigate about the matrix effect. For the recovery, it is to ensure test performance and maximize test sensitivity. Farms can reduce the effect of the hematocrit, so it can enhance the recovery. The use of FAMS tends to have less matrix effect, and the sample volume is less, and processing the sample to extract analytes has more steps. So for the future perspective, the use of FAMS for clinical trials of drug development and TDM during COVID-19 pandemic will be very useful because of its many practical benefits for clinical aspect as well bioanalysis aspect. As an effort to reduce indiscipline, analysts can use training videos or instruction brochures to socialize and train volunteers or patients on how to take samples with FAMS correctly. In addition, remote monitoring can also be applied by telephone contacts, online forums, or reminders. The author suggests that if one day there is an outbreak that requires further quarantine or an event that does not allow researchers to meet the subject, this FAMS technology can be applied for the continuity of clinical trial protocol for the drug development and also the TDM, considering its advantages. So the conclusion from our this review, the FAMS approach allows for sample collection at a location far from the clinic and can be done by the patient at home. This can be applied to the implementation of clinical trials for drug development and TDM during the COVID-19 pandemic, which requires quarantine to be held. In the aspect of bioanalysis, FAMS can improve the accuracy, precision, and recovery of the analysis by minimizing the effects of hematocrit. However, FAMS has concern about the stability of the analytes, so that special attention is needed to pay attention to the stability of these analytes in the FAMS sample. So thank you very much for your kind attention. Now I give back the time to the moderator. Thank you.
Oh, thank you. Thank you for your presentation, Dr. Yadiana. I'm sorry. So, yeah. So, do we have any questions for Dr. Yadiana? So,
No question.
Yeah, I guess there are no questions, Dr. Yadiana. It was really informative. Thank you for your presentation and your time for
Okay.
presenting. And yeah, thanks a lot. So,
Yeah.
moving on to our next presentation, we have Dr. Anamika from Jawaharlal Nehru University, India. And the title will be Positivity During COVID-19 Negativity, an optimistic vision for a better India. So, yeah, Dr. Anamika, so are you available? Yeah, Dr. Anamika, you can just click on the button screen. Yeah, that's perfect. Yeah, doctor, you can actually start your presentation. Please click on the button mic so that it'll unmute your thing. We're actually not able to listen to you. Your voice is actually not audible, Dr. Anamika.
Um,
Yeah,
can you hear me
now
now?
it's audible. Yeah, now it's audible.
Okay, just wait for
Yeah,
a second.
sure.
Yeah. So now the screen is very much clear to everyone.
Yeah, it's very clear. That's
Okay.
perfect.
Um, so good morning, good afternoon, and good evening, because all are in different time zones.
Mhm.
So this side, Dr. Anamika, and I'm currently working as an assistant professor at Jawaharlal Nehru University, New Delhi, India. I am a pharmaceutical scientist as a in background, and worked with different KPOs and Novartis Healthcare for about 13 years from now. And that is all about me joining the academic field. And currently, I am working in science policy towards improvement of India, and as a think tank for Indian government. So today we are going to discuss about a few things about positivity during this COVID-19 pandemic, which has affected each and every person world over. So what we have seen during these 6 months of COVID-19, and what India has emerged during this time with new solutions after the new challenges that has been faced by India. So some of these things we have seen world over, and some of these things are particularly pertaining to India. So the disclaimer informs that the views, thoughts, and opinions are personal of Dr. Anamika and nowhere related to my university or working institute. So COVID-19 is a Hydra-headed monster which has knocked the doors of infection world over in December 2019, where started from Wuhan, China, where the first infected patients was diagnosed. And later it has spread to other parts of the world and infected a hell lot of number of patients. Um, Initially, it was not considered to be a worldwide or world spreaded pandemic, but around January 30th this year, the WHO declared it as outbreak and health emergency of international concern, which is the highest level of alarm a WHO can raise, and ask the countries world over to take a control of the spread of the disease all over. And by the beginning of April 2020, the WHO has already reported 1 million cases of COVID-19, and which was about 10-fold increase in less than a month. So understanding of the pathophysiology of this coronavirus, its short and long-term impacts is still a mystery even after 6 months. We have understood, we are unfolding its structure, its pathophysiology, its impacts one by one, but still we are not 100% sure of what it is, what its long-term effects would be. If the vaccine comes, then would it be one time or for one time for the life, or we would require every 3 months, a booster dose of every year? We are very unsure about all these things. So we are scientists are really working hard to get into it. Further, on daily basis, we hear new things about COVID-19, including different types of symptoms. Initially, we knew about fever and cough are associated with this virus, but today we have come across a range of symptoms, including headache, rashes, conjunctivitis, loss of taste or smell, discoloration of fingers and toes, even diarrhea and cardiac arrest in few, and pneumonia is one of the major side effects. So there daily we come to know about new things about this virus, so which is a matter of concern world over. The treatment of this infection is yet not available, even after a long time. Being in a pharma industry, we all know that treatment of a particular disease, which is very new and nobody knows its pathophysiology, it becomes really difficult to work out on the treatment, especially when it's a viral infection. Bacterial infections are really easy to treat, but virus infections are always a difficult treatment for viral diseases. So researchers are working hard to bring out any vaccine or the appropriate treatment regimes. Although different types of treatments are said to decrease the effect of this coronavirus, but still all these treatments are majorly symptomatic. So there is no definite treatment regime that can be followed to treat this infection. There are only preventive measures which needs to be undertaken to break the chain of transmission, which includes sanitation and safety measures for the non-infected and the isolation of the infected persons. So then only the chain would be broken. And the future of mankind would be gloomy if the transmission of COVID-19 remain steady like this, which is happening currently. And the increasing number of this infection is really a matter of concern. And because of this, the different governments world over wanted to have lockdowns at different times according to their country economy. But we can see that it was a tough time for everyone when each one of us had been bound to stay at home. Even if we go out, we always have to take a lot of precautions and are at risk of getting infected. And government has to take strict action to break the chain of transmission. But now, as the GDP of the countries world over are soaring, we are proceeding towards unlocking of the countries. And now the responsibility is on the shoulders of the public so that they can take proper precautions while moving out for their work, for their daily groceries, or any other thing which is required for them to move out of the homes. Now we'll see what is the extent of spread of this coronavirus world over. This iron virus has affected about 31 million population with death of more than 9 lakh patients. And an unfortunate state is that United States, India, and Brazil are the three top most rankers, contributing to about 54% of the total infected patients, which is a very big number. To the World Health Status, about 50 to 60% of the world population will get infected with this virus once in their lifetime. So that is a matter of concern. Is a weapons, but it's a biological situation where we all are at risk. It's not not according to the geographical location of the world, but all over the world we are at risk. So we have to take a lot of precautions for this. As we have seen, but on the other side, as we have seen in the history, that world wars or other pandemics, besides causing destruction, also have opened up the doors of various inventions and innovative ideas that we are still using till date. We should understand that there is some positivity also in the tragic time of negativities. Number will help us understand the effect of the current outbreak. There is a reason that we are hearing more about negative news during this pandemic because of rise in connectivity all over the world. During earlier pandemics or world wars, the countries were not connected, but they were divided due to the absence of availability of internet and computer systems. So across the social media and the news, which are spreading the negativity of this disease, is the main reason that we are much more concerned. We are much more living in a much more worried environment, as was not the case with earlier pandemics or the world wars. I'm not saying that this disease or infection is not a matter of concern or worry, but we also need to see the other side of the coin, where we should admire the positive things which are happening around us. Let's see the numbers. According to the WHO, 80% of the COVID infections are only mild or asymptomatic. While 15% of the patients experience severe infections that might require oxygen, and only 5% of the total infected patients suffer critical and severe symptoms requiring ventilation and ICU care. With the increased number of treatment options, although not definitive but still symptomatic treatments, has led to the movement of this 5% bucket to 15% or even to 80%. So this number has been reducing day by day. So let's see what is the situation in India. Although the situation in India is grave, with about 1 million people being 1 lakh people being getting infected every day, but on the other hand, about 80 to 82,000 people are recovering each day as well. So let's see about the data till September 16, where we had about 50 lakh people who were infected with this deadly corona. I would not say deadly because it is causing death, but not to that extent. That the recovery rate was 78.5%, which is a good number with a mortality rate of 1.6%. I know that life of even single person is very important to be taken care, but this is a pandemic where nobody can control. But still, with the availability of the current treatments, we have reached to the stage that earlier the recovery rate was 15% in India, and then now it has rose to about 78%, which is a good number. And this indicates that majority of the infected patients will overpower this coronavirus in future as well, and only about 2%, or I mean less than 5%, will have to fight with this to emerge as victorious. So amid the COVID-19 pandemic, when the global economic superpowers are struggling to revive the economies from shock, India once again showed its power of resilience through never say die spirit. About 1.3 billion strong nation, where youth are in majority, are ready to take the baton of economic progress through their ideas of innovation. And during this mandatory on unwilling lockdown, we have seen a lot of positive things happening around us, including futuristic schemes launched by Indian government to cope up with the pandemic. And I remember when the initial lockdown of 21 days was announced in India, it was everybody was confused, and it might have happened in every country. That when initial lockdown was announced, everybody was confused how they'll be able to stay within their houses and lockdown, house arrested. But in these 6 months, people have started looking at the positive side of this lockdown as well. And as said earlier, wars of pandemic come with opportunities to improve. Improvement was seen in various fields across the country, and this is the thing we are going to discuss about in next few slides, how these improvements or the areas of expansions have been seen in Indian population. So what we have, so there are few areas which I've highlighted to discuss here on this platform. Where are the opportunities of growth, or where we have already seen expansion, like upgraded healthcare system, online education system, work from home opportunities, family time, quality family time, revived nature, different kind of learning opportunities, made in India businesses, reviving our culture during this lockdown time, diversification of work we have seen during this pandemic, and physical fitness along with reduced accidents. So what we have in improved and upgraded healthcare system? As we all know that COVID-19 is a major global crisis with far-reaching implications on our healthcare system. The main system which was affected by this was a healthcare system world over. And in India, it was a system which was never considered as a robust system. But as the coronavirus started knocking doors of different countries world over, Indian government started their groundwork by improving the level of healthcare and upgrading the healthcare system, increasing the ventilators, outsourcing the medical needs they required, making more ICU beds available for the patients, transforming different areas and creating temporary hospitals to address the healthcare needs of COVID-19 patients. So all these things happened, we have seen during this lockdown period. There was very initial stage when the lockdown was announced in India. It was to slow down the speed of transmission of infection and bought some time for health infrastructure to meet the challenge posed by the pandemic. And we all should not forget this thing that this improved healthcare system will not fade off with time, but obviously virus will fade off, and the Indian healthcare system will be able to tackle other viral outbreaks in a better manner in future. Then we had telemedicine. So I don't know how many of us have ever imagined that we can meet the doctor just over the call, and we don't need to visit the hospitals or the other healthcare facilities to get the basic consultation. This is really a stress buster for our senior citizen patients who need a regular checkup with their doctors. But on the other side, these senior citizens are really at a high risk of getting infected, and the death rate is also high among senior citizens for this COVID-19 infection. So we don't want them to visit the healthcare facilities. So telemedicine has really given them an opportunity to consult their doctors without visiting the healthcare facility. And also, this telemedicine is available 24/7 to cater the needs of the patients, where they can conduct the healthcare experts can conduct the preliminary screenings through online mode and guide them the appropriate actions, whether they can stay home, they can change their medications, or they do have a need to visit the healthcare facility. In India, healthcare workers have been trained to provide the telemedicine and reduce the patient visit to hospital and reduce the chances of infection transmission. Then we have Make in India business opportunities, the growth of Make in India products. So during this pandemic period, we read the media that India made ventilators and PPE kits, which are now available to cater the needs of our healthcare warriors all over the world. So during this lockdown, India has really grown its PPE kit and ventilator industry. And it's not just catering the need of healthcare workers of India, it is taking care of healthcare needs all over the world and supplying the ventilators and PPE kit to large number of countries. Now we can look at this at the numbers. As per the Ministry of Home Affairs data, of till August 2020, PPE kit industry has swelled to become 7,000 crore industry, starting from zero. India was not a manufacturer of PPE kits initially, and now it is delivering PPE kits to 5 other countries. Similarly, India manufactured about 60,000 ventilators in just 3 months, with the support from many established private sector companies, showing India's capability to overcome any crisis situation. So we have seen that such things now have established will not fade off, will remain in India. And we'll have made in India, make in India products. So this time of emergency has led to growth of Indian skill set and made in India products. Further, many companies are now looking towards India to set up their manufacturing units and invest in Indian market. So that's a boom. That will increase the cash flow to India and also the job opportunities to many young people. And Indian government is working hard to promote the house skills of Indian workers and Atmanirbhar Bharat, which is, and also boosting the Atmavishwas in the young people of India, making a self-reliant India. For this, the government is taking many steps to ease the flow of money through easy loans for startups and entrepreneurs. Here I am listing some government initiatives which the Indian government has taken to boost the economy, because there was a fall in cash flow among the country, because lots of people lost their jobs, lots of businesses were closed, small size businesses especially, because of the lockdown there was no business. So the government has taken a lot of steps to boost up the economy and announced about 1.3 lakh crore financial package, especially focused on emergency cash transfers to the poor. So that would take care of their daily needs. Further, it has announced about 20 lakh crore economic stimulus package, which is about 10% of the total GDP, and gives a clarion call for Atmanirbhar Bharat, or self-reliant India. Further, the government stressed on the 5 pillars to sustain the growth through economy, infrastructure, system improvement, vibrant demography, and demand. Government work to provide relief for poor, migrants, and distressed, involving free food, grains, cheaper loans, affordable housing, big boost to small industry businesses through, again, a big stimulus of about 6 lakh crores. The discussion will be incomplete without highlighting the efforts of the governments towards upliftment of an Indian youth through skill development, as the country is going to have a large youth population in the next 2 decades. So that is particularly important for the government to take care of the youth, which would be jobless if government doesn't take any step. So government has started programs like Skill India campaign, which is targeted to bridge the gap between lack of skill training and joblessness. Moving further, which was opted by each and every country world over, is the online education. It's not that nobody was aware of online education system. Everybody associated with the education system was aware of online education concept, but they never felt a need to implement it or were hesitant to adopt a new technology. So it's always a case. Whenever we feel that it's going on, then let it be. But if we feel there is a need to change, then only we change it. So this has happened during COVID for education as well. When there was no other option, then we had to opt for the new system of online education. And then everybody was, now by 6 months, everybody is well versed with the technology and has implemented it in every aspect of education. Every educational body, including schools, colleges, private coaching institutes, universities, have now implemented education through online mode, which is very convenient and saves time of commutation of students as well as teachers, requirement of less of infrastructure, and not only education, examinations are also happening online, and everything is a part of day-to-day education system is now based on technology. And now, again, highlighting the same thing, that it's not that online education system or the use of technology in education is going to fade off with time. It is the role of technology is going to increase in education system in future as well. The National Education Policy 2020, which was rolled on 26 July this year, highlighted the role of technological aspects in our education system, as and government has given a clear roadmap to move forward in this direction. And this revised policy has stressed upon the fact that all students, irrespective of their place of residence, should get quality education with particular focus on historically marginalized, disadvantaged, and underrepresented groups, as education is the best overall leader. So all these categories of students which were earlier deprived of the basic need of education will now be connected to education through technology. And there are diverse ways technology can be utilized to bring this transformation within existing Indian education sector, and has been clearly highlighted in the National Education Policy, which will be implemented soon in the education system of India. Another thing which we have seen in India during this lockdown period is work from home. Work from was never a part of culture of Indian workspace, even for IT companies, although this was common in other parts of the world, such as US and Europe. In the recent years, I should say about in the last decade, some companies have started giving work from home um, once or twice in a week, or 5 to 6 days in a month to their employees, but full-time work from home was not a culture in India. So with the advent of sudden lockdown, there was no other option for the companies to go for work from home culture. And this was not the option of only private sector companies, but government sector as well. They have to let the machinery work. They can't stop the machinery altogether. So they had to give the option of work from home to their employees. And now work from home culture has given a new vision of working, as more and more people can be hired companies with less infrastructure. So they don't need a 10,000 people capacity buildings to be constructed. They can just hire a person with a laptop and internet connectivity. That's all. So there has been changes in the mindset as well of the private sector companies. I know it will take some time in the government sector organizations to implement the work from home in future as well. But in private sector companies, now they are opting, even the country has moved towards unlocking, but they are still opting for work from home culture promotion for their employees, for their safety and ease and their comfort. And recently, a big MNC has announced that by 2025, about 75% of its employees would be working from home, so that they can manage their personal and professional life in a balanced way. So, this is one of the best part we have seen during this time. I worked with Novartis for so long, and I myself was working for from home for past 6 years. So this is where we can increase the employability of the young generation without compromising on other criteria and with less infrastructure cost of the companies. Now, come to the family time, quality family time. I would say quality lockdown has given us the option of quality as well as quantity family time. So before the spread of coronavirus, one of the rarest in a household was all family members relishing their meals or playing indoor fun games or enjoying a movie on a home theater together. So this pandemic has given us an opportunity to spend a quality as well as quantity time with the family. And companies are also offering work from home and flexi working hours, so people are able to balance their personal and professional life in a very better way. And not only this, people are now connected with old friends, old other far living relatives through social media, phone calls, which was difficult earlier due to different reasons. I mean, living in when you are staying far away from your office place, it's take really long time to travel from your home to office and back from office to your home. So this all has saved your time, and now even after working for 8 to 9 or even 10 hours a day, you still have some time for your family to connect with your relatives. And this is like one thing which has work from home has given to us. Moving further, we have revived nature and reduced pollution. In global summits, we keep on talking, we keep on discussing that our nature is deteriorating day by day. And it's just not in India, it's world over that nature is showing its effect by melting of glaciers, increase in temperature. But with this lockdown, mandatory lockdown or unwilling lockdown, there has been a reduction in pollution all over. And the nature has really revived because of less commutation on roads, less pollution from the factory outlets. So there has been a recent study that shows that River Ganga in India is reached to a clean quality which has never happened in past decades. It was a target of Indian government to make clean, but it was but never achievable. Now with less people visiting Ganga and throwing trash into the river, it has improved a lot. Water pollution improved, and AQI has reduced to about 50% of its original level, which was supposed to be in Delhi, for example. During this time every year, we used or 360 AQI, but currently I can see I just saw yesterday's newspaper and it was showing 140. So which is a great achievement compared to the Mandarin sounds. This pandemic period when we have time, we got the chance to revive our values as well and imbibe them into our next generation as well. So we have seen people coming from different backgrounds lending helping hands to those who are in need. So and then a decision of the Indian government to telecast mythological series was very well accepted by the general public and are helping children and young people to learn and understand more about our strong culture and enrich our lives. And it's not just that they are historical or mythological series, they don't have any relevance in today's time. These mythological series are really associated with learnings which are required these days to bind the family. Like Ramayana, if you have heard it, then it gives you a lot of learning values like respect your elders, keep your promises, and all these things like protect the weak, know the right from wrong, destroy the wrong. I mean, all these things are there which help us to revive our culture. and understand our values. And it's not just like we need values for Indian youth or Indian children, but it's required world over. So this has given us the opportunity to achieve that. And as we discussed earlier, that there are reduced traffic over the roads, and less people are using commutation methods like railways. So it has led to reduced number of rail and road accidents. And Mumbai, where we have local trains which are running across every minute, it has highlighted in a recent report that there has been a drastic fall in number of deaths over there. And it has been observed all over the country in India, and it must be from the world over. And even after factoring in pandemic-led deaths, the overall number of death claims for the life insurance industry has dropped by 20% in 2020. So this is a good thing. We have observed that as people are more and more people are working from home, they have to travel less, and otherwise also people are less traveling to meet their relatives or for any other purposes, so which has led to drop in rate of life insurance claims. Then we have different learning opportunities. What do you understand by learning opportunities? Learning opportunities is that besides our otherwise education, this lockdown has given us the opportunity to work, to increase our knowledge, to read our favorite authors, and also to adopt our hobbies, which we had lost due to our work schedule. So now we can revive our hobbies as well during this time. Like for me, I'm learning a new language. So this is to rediscover all your passion, because now we all are working from home. We can spare some time. We can and it's just not me. I mean, I sat with my son and we both try to learn a new language. So this is how the things are changing, and all this work from home is giving me the opportunity. And many digital platforms are even offering free ebooks to download and read through during this lockdown period. And companies are also offering virtual trainings through digital mode to inculcate new skills in their existing workforce. Moving further, we have diversification of work. Now, the people who were working on one field in their career for a long time, now we have seen a diversification in the type of work they are doing now. It's because the need of the hour, or it's just for helping others, or I mean whatever the reason may be. But now, for example, hoteliers are resetting up their hotels to use them as quarantine centers. Railway bogies are worked upon to isolation wards. Railway workers, policemen, army persons are working day and night to create masks and PPE gowns for our healthcare workers. So migrant workers who are stuck away, this is very interesting report I read, that migrant workers who were stuck away from their houses and are kept in quarantine camps are offering their skills in cleaning and painting out the quarantine centers. And all are coming together and working hard with the government to have a safe and better world tomorrow and be victorious against coronavirus. And this is last but not the least, that during this pandemic and lockdown time, fitness has become both a matter of concern as well as a necessity. Now we can't go out to gym, we can't go out for cycling or for any other workout. We need to do everything in our homes, but we need to be fit, because being not fit increases the chances of you being getting sick. So people are working on their physical and mental fitness through yoga, meditation, and exercise to overcome the negativity of depression during this disease time. As a lot of negative news we are reading about in blogs, on social media, on other ways. So we have to come out of this negative depression. So people are improving their immunity, which is mandatory in corona, that you should be strong immunologically. So through physical and mental yoga, they are doing that, and that also yoga has another plus point that we can do it in our houses. So we don't need to go out for that. People are turning on online workouts after global pandemic forces gym closures. And companies like Fitbit and other companies are also offering online workout training programs to their joinees, and probably providing different offers so that more and more people are inclined towards their fitness. In the words of Swami Vivekananda, any society can be made resilient through the process of man making. So, remembering our glorious past, the youth and wise men of the nation have always come forward and took the society to the path of progress and positive development. So, in the past also, we have solved many problems which were looking improbable to solve. As conceptualized by Pandit Deendayal Upadhyay ji, that only economic democracy, that is work for everyone, can only pave way for the development of the nation. Through the Atmanirbhar Bharat Abhiyan, the vision of $5 trillion economy looks a possibility through the economic democracy. The idea of Atmanirbhar Bharat can only become a possibility through the idea of oneness, which means oneness of ideas, oneness of vision, oneness of mind and effort. This will lead the nation towards glorious future. All these initiatives and development during these recent years will bring out the best in everyone. As with other pandemics and world war, this pandemic will also fade off, but new India's sun will rise with brilliance. M Health, M Education, E Office will be the bright future. And having full faith in scientific research community, we are hopeful that the time is not far when we have a success in getting a vaccination and other treatment regimes available to fight with this coronavirus. Any question and answers?
Oh, yeah. Thank you for your presentation, Dr. Anamika. So, do we have any questions for Dr. Anamika?
Even if we don't have them right now, here is my email and my LinkedIn details. If anyone wants to contact, please do so.
Sure. Thank you for your details. Yeah. We'll definitely pass this information to all the attendees right now, like present today. So, thank you for your presentation. Now, moving on to our next presentation, we have Dr. Aliasgar Shahiwala from Dubai Pharmacy College for Girls, UAE. Dr. Aliasgar, you can actually click on the screen button, yeah. You can just open your presentation, yeah. Please please click on the mic button so that you'll be able to talk and your voice will be audible to us. You are being muted from your side. Doctor, your voice is actually not audible to us.
Yeah, good morning, all of you.
Yeah, a very good
I hope
morning, Dr.
I
Alex.
hope it's still morning there because here it's around 8 p.m. in night in Dubai.
Yeah, it's around 12:10 p.m. here right
Okay,
now.
so yeah, good noon, and
Yeah,
thank you very much, the organizers, for giving me this opportunity to present my work
mhm.
and share my views at this
We're really
platform.
glad to have you here. Yeah,
Thank you so much. So I would like to move on straight
yeah,
forward to my presentation. All right, just a minute. Yeah. All right. So today I'm going to talk about some simple, very simple drug delivery technologies for intended therapeutic outcomes. Ultimately, why we are giving drugs or delivery systems to achieve intended therapeutic outcomes in the patients. And these days, there is too much overshooting in the technology. Lots of technologies are there. People are misusing technologies. So here in this presentation, I'm going to cover very simple and basic delivery technologies, using which you can also achieve intended therapeutic outcomes. As you know, what is the difference between the drug formulations and the drug delivery? Drug formulations is just formulating a drug in a dosage form to administer to the patient, while delivery is the next generation of drug formulations, which works actually to either enhance drug absorption or efficacy of a drug, or to reduce the side effects that is caused by the drug. Now, how they can do this? They can either control the rate at which the drug is released in the body, or the location at which drug is released. Some of the delivery systems, as we know, controls the both. So what are the main drivers for this new technologies in the drug delivery field? So mainly, basically, there are two drivers in the drug delivery field. On one hand, we have increasingly challenging molecules, while on the other hand, we have the increasingly challenging markets. So on the molecular side, the pipeline is full of molecules with lots of issues like viability issues, stability issues. We require target delivery, we require control release, especially with the emergence of biotechnology-based products. We have lots of manufacturability challenges. And from the other side, from the market perspective, there is increasing demands from the patients for reduced pill burden, improved palatability or convenience or taste of the medicines which they are taking. So current challenges any formulation scientist is having is summarized into these points. Sustained release of highly soluble drugs, which is difficult. Formulating increasingly water insoluble drugs. Mostly new molecules are synthesized. They are mostly suffering from water insolubility. So it's challenging for a formulation scientist to formulate increasingly water insoluble drugs. Also, to stabilize proteins and the peptides is a challenging task. Decreasing side effects and increasing efficacy of the current medicines by targeting. Usually we achieve by nanocarriers. Also, it is challenging to improve the patient convenience and to move from invasive to non-invasive drug delivery. So what are the solutions? What is the solutions to these challenges? As Albert Einstein said, if I had an hour to solve a problem, I had spent 55 minutes for the looking at the problem and only 5 minutes thinking about the solutions. So what I observe is in the drug delivery field, we have not spent enough time to define the problems facing the current challenges for developing clinically viable drug delivery systems. Thus, many of us are prone to stick to the what we are familiar with, even if they are not necessarily the best approach for some of the problems. So what we are doing actually, what is a common academic approach? So suppose if I see the publication of a drug, any new drug coming into the market, I see the publication with the liposomes, I will just change the system to move ahead with my publications. I will change it to neosomes or microspheres or sustainabilities or nanoparticles. Different. I'm just trying to fit the drug into different dosage forms. Or other hand, if I see any delivery system like liposomes, I'll try to fit different drugs into the delivery system. This is a very common approach. On the other hand, we move toward very complicated approach, which means we often innovate for innovation's sake. And this has led to technology overshooting, which simply makes technology more complicated. Here you can see in this figure, a nanoparticle is there attached with so many things, with radioactivity, with targeting moiety, with therapeutic moiety. So what's happening actually on the left scale, it's very easy to make this kind of systems, but when you want to transfer to the commercial scales, it fails. So when actually it is required to give benefits to the patients, it's failed to deliver so. And why we are adopting these academic approaches? Because of the increased pressure for publications, either for our promotions. There is a lots of competitions between the academicians, and the degree requirements, especially for the masters and the PhD students. So what is actually needed? So for my point of view, innovation in the drug delivery scientist has to be accompanied by intended ultimate effects. So we have to always start with the end in the mind, as clearly mentioned in the 7 habits of highly effective people. That is our ultimate aim, to treat or prevent the disease. Now, the problems such as cancers, diabetes, and other diseases can be corrected only after innovation is translated into clinically viable products, and these products need not to be complicated structure. And I'm going to demonstrate this by 2 of my case studies, 2 of my research work, how we can address the actual need and not just formulating new things without any tangible benefits. So these are the 2 case studies I'm going to discuss today. So first case study, in this case studies, I have taken the plant-based products or phytochemicals. As we know, 60% of the drugs are mainly originated from the plant sources, and later on it is modified through the chemical synthesis. So plants are the rich source of the medicines since the ancient ages. But these plant phytochemicals are suffering from either the poor solubility or poor efficacy or lack of targeting efficiency. So some of the issues I'm going to address in this presentation, which we have done in our lab at Dubai Pharmacy College. So this is the first study which was about improving the plant extract solubility as well as the efficacy. Now, this Fagonica indica is a widely available plant in nearby vicinity of our college, because as we know, the Dubai is a desert basically. So this is the plant which belongs to this desert of the UAE. It's also grown in the other parts. And this antioxidant and hepatoprotective activities of this plant extract was already established earlier by one of my colleague. Her name is Dr. Najla Sehab, and she has already published, and the reference is given in the slide for that. Now, the challenge was, she came to me to further enhance solubility and or bioavailability of the extract, because as we know, the extracts are mainly the alcoholic in nature and they suffer from the poor solubility, and solubility is the first and foremost criteria for the bioavailability of a drug, because only soluble materials can cross the gastric wall and enter the portal blood circulation. So looking at the different approaches which can improve the bioavailability, I came across this. So either we can use surfactants, we can use cyclodextrins, nano or microsuspensions. We can also use co-solvents approach. We can use salt formation approach, and we can also have lipid-based formulations like microemulsions, self microemulsifying drug systems, and so on. But out of these approaches, what is the simplest approach and which is the most efficient approach? So we decided to go for solid dispersants, as solid dispersants provides one of the very simple approach which is effective and economic for improving dissolution as well as the bioavailability of poorly soluble drugs. Basically, it's a dispersion of active ingredient with the inert carriers. Now, based on the carrier use, the solid dispersants are classified into first generation, where the crystalline carriers were used like urea or sugar. Later, second generations of solid dispersants were synthesized, which includes amorphous carriers, usually polymers like polyvinyl pyridone and polyethylene glycol, which are the most common polymers used. And later, third generation are also invented with addition of some surfactants or surfactant active agents along with the polymers. So we decided to start with the second generation because first generation solid dispersants are thermodynamically more stable and do not release the drug easily. In the second generation, we are supposed to use amorphous polymers, so we choose most commonly used and most widely used and the cheaper options, which is the polyethylene glycol 4000 and polyvinyl pyridone K30, because they are also very highly water soluble and they are regarded as safe. So with these two polymers, we synthesize our second generation solid dispersants, and here are the results. If you see the solubility of the extract, which was around 200 microgram per ml, which was increased to around 260 with PVP when 1:2 ratio was used, and which was increased to around 270, 280 or 290 with PEG 4000. So we decided to move with the PEG because PEG was providing the better solubility. However, on further increase on the ratio, there was not much increase in the solubility was found. So the ratio optimized for the second generation solid dispersion was 1:2. So with this, still it is not a very good enhancement in the solubility. So we decided to further improve by moving onto the third generation. So we incorporated surface active agent into our solid dispersion, and different surface active agents were tried like bridge, twin 20 and twin 80, and out of that twin 80 was found to be the best one, providing twin 20 was found to be the best one, and the 50 milligram with the ratio of 200 milligram of extract, 350 milligram of PEG 4000 with 50 milligram of twin 20 was found to be the best in increasing solubility. Beyond increasing to this limit of twin 80, twin 20, we found that the mixture was losing its consistency and it becomes like more liquid in nature. So we decided not to go further and take this as an optimized formulation. Here are some of the results for particle size. If you see, the particle size was found to be around 1.9 micrometer with polydispersion index of 0.425. Sorry, the left side was the left side graph shows the diameter of the plant extract, which is 1.972 micrometer, while on the right side it is showing the you can say the solid dispersions particle size, which reduced to 437.6 nanometer. So which indicates not only increasing the solubility, but also there is a reduction in the particle size of the plant extract due to the solid dispersion. So then we have come to the efficacy evaluation by doing anti-hepatotoxicity activity of the extract along with the solid dispersion at 2 different doses. So the suspensions of the extracts were prepared in 1% CMC, and paracetamol was injected because it is the hepatotoxic agent in 1 gram per kilogram into the rats, and then the rats were divided further into 4 different groups according to the treatment, 2 different doses with the plant extract and 2 different doses with the solid dispersants, which are equivalent to the same doses of the plant extract. Sorry. Yeah. Then the blood samples were collected and the level of liver enzymes like ALT, AST, ALP, total bilirubin and total proteins were determined. And these are the results which were obtained. So here, if you see the results, the first group is the normal group. If you see the paracetamol treated group, which is showing the very elevating levels of the ALT, ALP, AST and total bilirubin, while the protein count total protein was decreased. This clearly indicates toxicity of the paracetamol. Now, when the plant extract, there was no much improvement in the plant, or there is a slight improvement due to the efficacy of the hepatoprotective efficacy of the plant. There was a slight improvement in the hepatotoxicity. While if you see in case of the solid dispersants, the levels of enzymes and the proteins are closest to the that of the normal at 500 milligram dose. This indicates that the extract in solid dispersion formulations clearly able to revert the toxicity induced by the paracetamol in rats. So what are the significant findings of this result? As we know, the alcoholic plant extracts are poorly water soluble, and suitable formulation strategy is required to improve its solubility and viability. Solid dispersion approach was adopted due to its cost effectiveness and ease of preparation without any complicated structures, and optimized solid dispersion formulation at 500 milligram dose, so complete recovery from hepatotoxicity induced by paracetamol in rats. And this was actually published in the Pakistan Journal of Pharmaceutical Sciences. Now, move on to the case studies number 2. But before going to the case study number 2, I would like to ask the moderator, is it do I have the sufficient time to move on, or my time is finished?
You can actually continue, doctor. No problem. You can take your time. No problem.
Okay, fine. So I'll try to wind up within, I think, 15-20 minutes. So
Yeah, no
here
problem.
I'm going
You
to,
can continue.
oh, okay. Thank you so much. Yeah, thank you. Appreciate it. Yep. So now second is about the cancer therapy. As we know, cancer is the third or second most deadly disease actually after maybe the accidents and I think some other disease. The cancer is the third most deadliest disease, and the treatment still the effective therapy is not available. And as we know, the plants have played important role as a source of effective anti-cancer agents and currently used like paclitaxel or docetaxel. These are mainly from the plant sources, as we know. So now, what is the difficulty with the plant extracts or the plant sources? They are lacking the targeting efficiency, or they are lacking the intake by the cancer cells. Means they are not able to penetrate inside the cancer cells. So our this research was conducted specifically to increase the targeting inside the cancer cells or the tumor cell lines. So we done the routine studies like standardization of the plants with respect to the British pharmacopoeia requirements. We extract active excipients like flavonoids and the phenolic compounds, which are mainly responsible for the antioxidant and thus anti-cancer effect. And we determine total phenolic and flavonoid contents of the plant extract by Foley culture reagent. So here, because we need to achieve intracellular delivery, the nanoparticles is the best approach. Because nanoparticles are usually taken up by the endocytosis, and which is not the saturated process, which is usually occurs with the normal solutions. So nanoparticles are again prepared with the chitosan, and the chitosan was used. Why? Because chitosan is naturally occurring cationic polymer, which is biodegradable in nature. And since the plant polyphenolic compounds and the flavonoids are negative charge, we utilize this property of those compounds and we choose cationic polymer. So basically nanoparticle formation becomes very easy. There is no complicated steps required. So they were nanoparticles were simply prepared by the polyelectrolyte complex method, or which is also known as self-assembled polyelectrolyte method. So in this method, chitosan was dissolved in acetic acid, and the extract suspension was added drop by drop to forms the and were homogenized to make the nanoparticles. And these nanoparticles were further congealed with addition of sodium citrate to achieve or to enhance the ionic gelation. So after addition of sodium citrate, we have obtained our chitosan extract nanoparticles. We did characterization. So first of all, we did thin layer chromatography to check whether there is the free polyphenolic compounds are there present in the sample which the nanoparticles which we prepared. So because the difference that the RF value will be different for the nanoparticles and the plant extract. So which is a very simple tool which is available. We have also characterized the interaction of the polyphenol compounds with the chitosan by doing the FTIR, and we also measure the particle size and the zeta potential of the nanoparticles by the zeta sizer nano zetas. So I'm not going to share the results of the TLC and the FTI here. I'm directly moving to the results of the size, particle size. As you can see here, the particle size of the nanoparticles are within 100 to 1000 nanometer, which approximately around 400 to 500 nanometer. And as you see, the zeta potential, which is positive around 40 millivolt, which is due to the presence of the positively charged polymer chitosan, and this zeta potential value, the high zeta potential value, also suggests the stability of the prepared nanoparticles. Then we conducted anti-cancer cell proliferation activities with the human breast cancer cell lines, and I don't go into detail because it is very common. And here are the results of the cell availability of different formulations used. We have used 5 milligram of 5-fluorouracil as a standard drug, and as you can see in this standard drug 5-fluorouracil 5 milligram, it is cell availability reduced to just 10% from initial 100%, which is very effective in killing the cancer cells. When you apply, when we expose the cells lines with the 25 milligram extract up to 250 milligram extract dose, we have seen some very little anti-cancer cell proliferation activity. You can see by looking at the cell availabilities, which are almost around 65%, and with 250 milligram extract, it was reduced to around 45%. But still, it was not very good achievement. So we also tested the blank nanoparticles because chitosan itself has some anti-cell proliferative activity. So we suggested around 72% cell availability in the chitosan, and then we tried the different doses of nanoparticles. This 0.1 mg, 0.5 mg, 1 mg, and 2.5 mg nanoparticles means these nanoparticles contains this much equivalent amount of the plant extract. It's not about the quantity of the nanoparticles, it is about the quantity of the plant extract, which is entrapped within the nanoparticles. And as you can see here, with 2.5 milligram nanoparticles dose, we are able to achieve the same anti-cancer proliferation activity, approximately same compared to 5 milligram of 5-fluorouracil or the standard drug, which was a very significant achievement of this study by using a very simple approach. So the significant findings were almost 500-fold reduction, that is from 250 milligram to 0.5 milligram in the extract concentration required to achieve the same anti-cancer cell proliferation activity when formulated as nanoparticles. As I mentioned, 2.5 milligram extract, similar activity to that of 5 milligram of 5-fluorouracil. And our results actually proved that the traditional medicinal plants could be an excellent source of natural anti-cancer agents, and the chitosan extract nanoparticles is a promising process strategy to enhance the clinical effectiveness of the plant extract. And this was published in Current Cancer Therapy Reviews. So before ending, I would like to share my thoughts. I strongly believe that we should make a better environment for researchers to provide or to do their work, best work, without worrying about the short-term productivity in terms of the publications. Since the drug delivery is really multi-dimensional research field, which needs collaboration from different players, And recently there is lots of role digitalized or role of AI, artificial intelligence is also coming up. And it need not to be very complicated structures or delivery systems for clinically beneficial formulations. And there is a very famous quote by the Albert Sazent, which says the research is to see what everybody else has seen and to think what nobody else has thought. And this is the place I work. Dubai is a famous city in UAE, and I think most of you have visited. And this is the college. It is the old building. The photograph recently it was renovated, and it is a very nice ambient is there. And these are some of the books which I have published. Now my upcoming book is on the applications of artificial intelligence in drug delivery with elsewhere. So I'd like to thank you all once again for giving me this opportunity and share my work. Thank you. And I hand over to the moderator. Thank you so much.
Yeah, thank you for your patience in presentation. It was really informative, Dr. Ali Sagar. So yeah, uh, now do we have any questions for this presentation? So yeah, I guess there are no questions, Doctor. So we'll just see maybe if anyone gets back to you through email, so you can just reply to them for sure.
Yeah, anytime, anytime. And I'm looking forward to the collaborations also. If anybody wishes to collaborate with our group, you are most welcome.
That's really great. And yeah, thank you for the opportunities. Yeah, we'll definitely pass this information to everyone through email also. So yeah, they can just contact you in any case if they need it. So
Really appreciate it.
thank you. So coming to our next presentation, it's actually from Dr. Sai Sailesh Kumar, but he's actually not available right now, and he has just left us a message that he has gone on an emergency meeting. So it's not possible for him to present today. Maybe if there's any possibility, he'll do that tomorrow. So we, I guess we are only left with the two presentations. So we are waiting for Dr. Shalini Tanwar and also Dr. Vamshi Krishna. But in the meantime, I just want to know if anyone is interested in taking a break, or we can just wait for a few minutes. So I'll just make a call to them, or we can just go for a break for 40 minutes and we'll return back for the lunch after the lunch session. Please let me know your opinion so that we'll just get back to you. We just received a message that Dr. Shalini is actually not available immediately. So what we'll do is we'll just go for a break of 40 minutes, and as the time is now 12:40 here, so we'll just return back around 1:15 after the lunch, and we'll continue with the other two sessions. I hope everyone is okay with this. If possible, please do let me know your opinion so that we'll just finalize this and we'll get back again. I do understand, like, we have different participants from different countries, but yeah, we'll just get back to you as the presenters are not available right away. So we'll have to wait for them and We'll get back to you again at 1:15 p.m. So yeah, we'll get back again at 1:15 p.m. Thank you all. Hi everyone. So we have a few of our delegates present right now. So yeah, I guess we can start for now. So we have our next presentation from Dr. Shalini Tanwar. Dr. Shalini, are you available right now?
I am not able to share my presentation. I'll need a few minutes to change my
Okay. Sure, no problem. You can once you're ready, please do let me know so that we'll just continue with this.
Sure,
Yeah, sure. Thank you.
no issues.
Dr. Shalini, it's actually visible right now.
Am I audible?
Oh, yeah, you're audible now. That's perfect.
Okay. Is everything
Yeah,
good? So
yeah, it's
may I
all
start?
perfect. Yeah, you can start now.
Let me. Yeah. Um,
Uh, yeah, if
okay.
in any case, if you're not able to view your full screen, you can just
Mm-hmm.
move the buttons. Like you'll just have four lines over there. You can just move here and there.
Okay.
Yeah.
Yeah, yeah, yeah. Mm-hmm. Yeah. Okay. So now it's good. Yeah. So is everyone ready? Shall I start?
Yeah, we can start.
Okay. So good afternoon, guys. My name is Shalini Tanwar. I'm a research fellow at NIH since 2015. And as I would agree with the last presenter before the break that we need for any drug to be dis- um, shunted out to the market, there is a lot that goes on behind the curtains. So my job is the basic biology aspect of the drug. How does the drug works? So it is both before and after the release of the drug into the market. So one such drug that I worked on in the major project for my postdoc research was type 1 interferon or interferon beta. Interferon beta is given to multiple sclerosis patients to relieve the clinical symptoms of the disease. So to give you the background, what is multiple sclerosis? Multiple sclerosis is a central nervous system debilitating disease. So basically, your own immune system starts attacking the central nervous system of your body, and because the nerves are being attacked, the person starts losing control over muscles and control over every activity. So the grip and everything, it goes down. And the other major feature of this disease is that it comes in waves. So there is a wave that will come, like the patient will have very severe symptoms for a while, and then the symptoms will continue to this will start improving for a while, and then the patient improves, but not like a completely normal individual. He will still have some permanent damage done to the nervous system, to the nerves, but for some period, and that varies from person to person, and we don't know how and why, but for that period, the person may continue to live a normal life. Then again, the second wave, and then a third wave comes. And with each wave of the immune system attacking your nervous system, the person just keeps on, the nervous system of the person just keeps on deteriorating. So interferon beta is given to reduce these clinical symptoms of the disease, the loss in control of your nervous system. And it has been shown that it is pretty effective in reducing the clinical symptoms. But the thing is that the receptor for interferon beta, it is called IFNAR receptor, type 1 interferon alpha receptor. IFNAR receptor is present in all the cells of the body. So you name it, epithelial cells, endothelial cells, fibroblasts, immune cells in themselves, like T cells, B cells, dendritic cells. So basically every cell of your body, it has the receptor for interferon beta. So how exactly is this drug reducing the clinical symptoms is the thing that the field is still working on. And there have been various studies in which it has been shown that interferon beta, it induces a Th2 immune deviation in the patients. So basically what happens is when your immune system attacks your nervous system, there are two types of T helper cells. Th1 and Th2. And Th1 via secreting interferon gamma and IL-17A cytokine attack the nervous system. However, the Th2, the T helper 2 immune system, the Th2 phenotype, is manifested by the secretion of IL-10 cytokine. And IL-10 cytokine, it actually um, suppresses or improves or reduces the Th1 phenotype, and thus it is said that if a cell does not progress towards an interferon gamma and IL-17 secreting, rather becomes IL-10 secreting cell, you will not have the cells that would target your nervous system and would debilitate it. So this is one way in which interferon beta acts. The other way that has been shown is that interferon beta, it downregulates the co-stimulatory molecules like MHC2 and other co-stimulatory molecules on your dendritic cells and antigen presenting cells. So now the T cells that would have attacked the nervous system cannot see the antigen with the co-stimulatory signal, and thus they won't be activated to attack the nervous system. Apart from it, interferon beta, as I said, is the IFNAR receptor is present on the nervous system also. So the IFNAR IFN beta, it also increases the nerve cell survival of even after being damaged by secreting nerve growth factor. So this is one more way in which interferon beta is helping the patients. So to delineate which is the exact and the main mechanism by which interferon beta reduces the clinical symptoms. So what the field did was they took PBMCs from the multiple sclerosis patients before and after interferon beta treatment, and then they did the RNA-seq analysis and various sequencing analysis to see which pathway pops up. And there wasn't too much drain, there wasn't any major pathway that came up in these gross PBMC stimulation sequencing studies. So in 2008, there was a paper that was published in Immunity. They used various conditional knockout mice models to study the role of interferon beta. So in this paper, what they did was, this is the IFNAR global knockout mouse. So in this mouse, none of the cells of the mouse will have IFNAR receptor. So whether the interferon beta is secreted or not secreted, it doesn't matter because the cells will not receive the signal, and thus this would, when these mice will be subjected to multiple sclerosis induction in mice, this is called experimental autoimmune encephalitis. So EAE induction, these mice will tell us which cells are responding and how are the cells responding, and then we can study the role of interferon beta. So here they saw that when these either global knockout mice or control mice, the wild type mice which have the interferon receptor on the surface of all the cells, they were induced EAE. What happened was that the mice that did not have any IFNAR receptor in any of the cells, they had much severe disease. So it establishes the role of interferon beta, that interferon beta does reduce the clinical symptoms of the disease. So on the y-axis is the clinical score, and on the x-axis is the number of days in which the disease is progressing after the EA induction. So after establishing that interferon beta does reduce the clinical symptoms, the next question was which cell type is more important. So here they used IFNAR floxed mice and bred them with either CD4 mouse or lysine mouse. So in these mice, what would happen? So IFNAR gene is floxed, and when you breed these mice with CD4 expressing mouse, so in all the cells in which CD4 will be expressed, the IFNAR receptor will be floxed out. So basically in this case, the T cells will not express the IFNAR receptor. Nerve cells, dendritic cells, everything else will have the IFNAR receptor on their surface. and then they induced EAE in these mice. And here they observed that though the mice that did not have IFNAR receptor on the T cells are the black filled circles, and the wild type mice are the open diamonds. So they observed that there is a lag in the time of induction or the setting of the disease. So in like the mice in which the IFNAR receptor was not on the T cells, they started having the clinical symptoms as early as 11 days in their case, while the wild type mice started expressing the similar symptoms as late as 13 days. So there was a delay of about 2 days if you have IFNAR receptor on the T cells. Apart from this, the major striking data for them was that when they used the mouse in which bred the IFNAR flux mice with lysin-free mice, so in these mice, the IFNAR signaling would be depleted from myeloid compartment. The myeloid compartment is your neutrophils and your monocytes. So if they do not have the IFNAR signaling, now this graph kind of mirrors this global knockout graph. So basically it says that IFNAR signaling on myeloid compartment is much more important for reducing the clinical symptoms of the disease via interferon beta. But we focused on this lag of setting of the disease when you do not have or when you do have IFNAR receptor on the T cells. So, and our focus was the T regulatory cells. Now, T regulatory cells are the branch of T cells that suppress the T cells, the activated T cells from or CD4 T cells from getting activated, or if they are activated, from secreting the cytokines. So what I did, I took the IFNAR flux mice and bred them with FOXP3 cre mice. So what would happen in these IFNAR flux FOXP3 cre mice, which I would in my subsequent slides call as conditional knockout mouse, is that these mice will have IFNAR receptor on the myeloid compartment. They will have IFNAR receptor on the CD4 T cells and the CD8 T cells, but they will not have the IFNAR receptor on the T regulatory cells, which are the major suppressors of the immune system. And these mice were then induced the EAE, which is the mice model for multiple sclerosis disease. And here I observed that as compared to the controls, which are black filled circles, the conditional knockout mice had much severe disease. So if at the same time when the wild type had the disease score of 1 to 1.5, the conditional knockout mice had a clinical score of about 4, 3.5 to 4, which is a very severe form of the disease. So it basically says that if you do not have interferon beta signaling going on in this suppressor population, the T regulatory cells, you have much severe clinical disease. So as I said, the disease is that the T cells start attacking your nervous system. So are they attacking the nervous system in this case? So I looked at the central nervous system, which is the brain and the spinal cord of these mice. And by flow cytometry, I found that in the conditional knockout mice, we had more CD4 T cells in the CNS tissue. And much more of these cells were secreting IL-17 cytokine. So now it has to be noted here that usually in the central nervous system, you do not have T lymphocytes. There is a blood-brain barrier which bars the lymphocytes from entering to the central nervous system, because as soon as they will enter the central nervous system, the CNS, they will attack the CNS by considering it as foreign. So in these mice, when the T regulatory cells were not receiving the IFNAR signaling, the T cells had breached the blood-brain barrier and were in much more proportions as compared to the wild-type mice. And they were secreting much more the cytokine that is responsible for the disease. So basically, it's saying that there is disease in the absence of these IFNAR signaling on T regulatory cells. So now, our question was whether during the priming stage, so if you see the death in the disease curve, you see that you have like about 7 to 8 days in which the mouse is normal even after the induction of the disease, and the disease starts being expressed at around day 8 in the conditional knockouts, and by around day 11 in case of wild types. So what is happening during this initial period of quiescence, which is not actually quiescent for the cellular phenotype? The T cells are being activated in the draining lymph nodes, and if the T cells are being activated, why are our T regulatory cells not being able to stop them from being activated, or stop them or suppress them from secreting cytokines? So when I observed the draining lymph nodes of these mice, to my surprise, I observed that the proportion of CD4 T cells in both the wild type and the conditional knockout mice was comparable. Even the activation status, barring the day 3, which is very earliest time point, at the later time point, even at day 5 and day 7, the proportion of activated CD4s were comparable between the two mice types. And though it is too early to look at the cytokines being secreted by the T cells in the draining lymph nodes, but even then, when we looked, the proportion of cytokines being secreted is equivalent. When we looked at the proportion of T regulatory cells, the proportion of T regulatory cells was also comparable in the draining lymph nodes throughout the priming phase. So basically, it says that the T regulatory cells are present at equivalent proportion in the two mice types. The CD4 T cells are present at equivalent proportion, and even the activated phenotype is equivalent between the two mice types. Even then, we have more disease in the conditional knockout mice. So why is it so? Are the T regulatory cells not being able to suppress? Are they not activated? So we looked at the activation status of the T regulatory cells by staining for the Ly6C expression on them. And here we observed that instead of, like, we would expect that the T regulatory cells in the conditional knockout mice are not activated. That's why they are not able to suppress the CD4 T cells. However, the T regulatory cells were more activated in case of the conditional knockout mice. Even then, they were unable to suppress the T cell activation and cytokine secretion during the effector stage of the disease. So we wanted to know whether these Tregs, even after being activated, are they stable or not? So to check the stability of the T regulatory cells, we induced EAE into our wild type or conditional knockout mice, and then at day 3, we sorted the T regulatory cells out from the draining lymph nodes and transferred them into congenic mice, wild type congenic mice. And after 18 days, the cells being parked in these wild type naive mice for 18 days, we looked at the spleen for these activated T regulatory cells. And we observed that the proportion of the T regulatory cells and the FOXP3 MFI was comparable between between the T regulatory cells that were receiving the IFNAR signaling or that were not receiving the IFNAR signaling. So basically saying that T regulatory cells, even if they do not receive the IFNAR signaling or IFN beta stimulus, they are pretty stable. They can get activated, yet they are not able to suppress. So we looked at various suppressor molecules being expressed by the T regulatory cells, via which they suppress the T cell activation. And these molecules are CTLA4, OX40, neuropilin, CD25. All these molecules were comparable between the wild type and the conditional knockout Tregs. So basically saying that T regulatory cells do not have a functional they are not functionally compromised in the absence of IFNAR signaling, yet they are not able to control the T cell activation and to control the disease being exacerbated. So we looked at the draining lymph nodes for other cell types that are present. And at the same time, we looked at the literature also for what are other cell types involved in EAE in mice and multiple sclerosis in humans. And here we observed, we realized that there is a population of myeloid cells called myeloid-derived suppressor cells, which are characterized for flow cytometry as CD11b positive, Ly6G positive, Ly6C low for granulocytic MDSCs, and Ly6C high, Ly6G negative for monocytic MDSCs. And the field is pretty um, iffy about the role of these cells in case of autoimmunity. They are still debating whether these cells are pathogenic or suppressive in case of autoimmunity, because as you may see here, that a study in Journal of Immunology 2007 says that CD11b positive Ly6C high cells, which would be monocytic MDSCs, they suppress the T cell proliferation by production of nitric oxide. However, CD11b positive, GR1 positive, which will be a mix of both these types of the MDSCs, it promotes Th17 differentiation, which is the IL-17 secreting CD4 T cells or IL-17 secreting Th17 cells, and thus are pathogenic in case of EAE. So first, it was important for us to establish the role of these MDSCs in case of EAE, whether they are suppressive or pathogenic, and what is the phenotype in our mice. So to establish whether they are pathogenic or suppressive, what we did was we took wild-type mice and induced EAE in these mice, and at the time of EAE induction, what we did was we gave a single shot of myeloid cell depleting antibody to these mice. And this single shot of myeloid cell depleting antibody did not have any effect on other cell types, be it CD4, CD8, CD11Cs, B cells, or NK cells, and which is expected. And it reduces the proportion of CD11B cells, that is the myeloid cells, by about 50% for first 5 days of the after having given the antibody. And by day 7, you again start having because these myeloid cells are being churned out from the bone marrow every 2 days, like all the time. Their half-life is every 2 days, so they are being churned out in a large number from the bone marrow at all the time. So by day 7, they make up for this loss of the myeloid cells that had happened during these 5 days. So we see that there is a transient reduction of myeloid cells with the induction of EAE. And what happens in this case is that this transient reduction of myeloid cells leads to much severe disease as compared to what we had observed in our mice. And it was almost similar to what had observed in the global IFNAR knockout mice. And the disease was so severe that about 50% of the mice, they actually died between day 5 to day 15. And this is the time when the activated CD4 T cells, they start leaving the draining lymph node to the effector site. So basically, the CD4 T cells were activated in such large proportions and were secreting so much interferon gamma and IL-17 that the mice were dying of cytokine shock. And this was because of transient reduction, not the absence, just transient reduction of the myeloid cells. So we went back to our conditional knockout mice and looked for these myeloid cells or myeloid-derived suppressor cells in the draining lymph nodes of our mice. And very surprisingly, we found that, so this is the classic definition of myeloid-derived suppressor cells, that you do not have any of MDSCs at day 0. Basically, in naive lymph nodes, you do not have MDSCs. As the inflammation ensues, you start seeing a surge of these cells in a wild-type mouse. But this surge was conspicuously absent in the mice that did not have IFNAR signaling on T regulatory cells. they did have IFNAR signaling on the myeloid-derived suppressor cells. But the absence of IFNAR signaling on T regulatory cells led to the absence of these additional suppressor population in the draining lymph nodes in the priming phase. Now, we wanted to see whether this is because these cells are not being churned out from the bone marrow. So we looked at the bone marrow of these mice during the priming phase, and the proportion of, yes, I must mention that this conspicuous absence was similar for the granulocytic MDSCs and for the monocytic MDSCs. So at this stage, we did not find any differential activity or differential status between the granulocytic and the monocytic MDSCs. So I will just say MDSCs for the subsequent slides. So in the bone marrow, we did not see any differential generation in the conditional knockout mice. So to see that the absence of IFNAR signaling on T regulatory cells does not inhibit or does not affect the generation of MDSCs in the bone marrow. So is it that the MDSCs are not being recruited to the draining lymph nodes, or they are not being recruited anywhere in the body of the mouse? So we looked at the spleen and the central nervous system, which would be the effector site of the disease during the priming phase, and we observed that all throughout the priming phase, while they were absent in the draining lymph nodes where the CD4 T cells are getting educated about the antigen, rest of the tissues of the mouse, they have these MDSCs. So even the CNS have the equivalent proportion of these cells throughout the priming phase. So it is a specific issue of recruitment of these suppressor population to the draining lymph nodes. Now, to establish that this is due to the IFNAR signaling on the T regulatory cells and not on the myeloid cells, I used the mouse that the earlier researchers had used, IFNAR flox lysine mouse, and induced EAE in these mice, and then looked for these MDSCs in the draining lymph nodes. And in the draining lymph nodes of these lysine mice, I observed the proportion of MDSCs was comparable. So this cements the fact that it is the IFNAR signaling on T regulatory cells that recruits the MDSCs, or that helps recruit the MDSCs to the draining lymph nodes of the mice during the inflammation. So how are T regulatory cells doing this? So usually T regulatory cells are shown to exert their effect either via secretion of IL-10 or TGF-beta. So I induced EA into the wild type mice, and at the time of induction, I blocked the IL-10 receptor of these one of the test group of the mice by injecting them with anti-IL-10 receptor antibody. And when I looked at the draining lymph nodes for the MDSCs, it was comparable. Even in the mice, which I bred these mice TGF-beta flox FOXP3 YFP3 mice. So in these mice, the T regulatory cells would not be able to secrete TGF-beta, and even in this case, the draining lymph nodes had comparable amount, proportion, and number of the myeloid-derived suppressor cells. So T regulatory cells are not doing this via IL-10 or TGF-beta, but how are they doing it? So for this, we went back to CD4 T cells. So usually T regulatory cells, what are T regulatory cells doing and how are T regulatory cells doing something is very difficult to find. However, we can see the effect on the CD4 T cells. And for that, what we did was we sorted the CD4 T cells from either the wild type mice or the conditional knockout mice after having induced EAE. And these activated CD4 T cells from these two mice types were subjected to RNA-seq analysis. And when we looked at the data of these after the RNA-seq analysis, we were very surprised to find that the CD4 T cells from the mice in which CD4 T cells can receive the IFNAR signaling but T regulatory cells cannot receive the IFNAR signaling. Even then, the CD4 T cells were defective in their secretion of chemokine ligand CCL8. I confirmed this using flow cytometry. So immunized the mice, we induced EAE into these mice. So this is the data. So in naive mice, this is the MFI of CCL8 in the wild type mice and the conditional knockout mice. So basically, the amount of CCL8 in the draining lymph nodes and or in the lymph nodes in the naive mice is comparable. In the wild type mice, as the inflammation ensues, there is an increase in the MFI of CCL8 by the CD4 T cells. So basically to say that the CD4 T cells are now secreting the CCL8 in response to inflammation in the draining lymph node. However, this increase of MFI or this induction of CCL8 is absent in the conditional in the CD4 T cells from the conditional knockout mice. And it's not just the CD4 T cells, the non-CD4 T cell compartment, which would comprise the B cells, APCs, they were also defective in their upregulation of CCL8. So we wanted to see whether it's just CCL8 or other cytokines as well, or other chemokines as well. And we did ELISAs on the draining lymph, on the sorted populations from the draining lymph nodes of either wild type mice, which would be the black circles, and the conditional knockout mice, which would be the red squares. So in all these cases, the CD4 T cells, CD8 T cells, B cells, and endothelial cells from the conditional knockout mice, though they could receive the IFNAR signaling, they were defective in their secretion of chemokine ligands CCL21 and CCL9, all because the T regulatory cells in these mice could not receive the IFN beta signal on them. So the entire study thus concludes that selective deletion of IFNAR on the T regulatory cells, it results in severe EAE because the myeloid-derived suppressor cells require the chemokine ligand gradient being generated in the draining lymph nodes. This chemokine gradient is being generated by the secretion from either CD4 T cells, endothelial cells, B cells, CD8 T cells. Now, if T regulatory cells are not receiving IFNAR signaling or interferon beta signaling, they are suppressing these T cells. Remember, the T regulatory cells were activated and were more activated and more suppressing. So they are suppressing the secretion of chemokine ligands by all the other cells, and thus by suppressing the chemokine ligand secretion, they are suppressing or blocking the entry of myeloid-derived suppressor cells into the draining lymph nodes. Now, since these MDSCs are not being able to enter the draining lymph nodes, they are not able to suppress the CD4 T cells from being activated towards this antigen, and thus you have the severe disease. Thus, this study demonstrates that T regulatory cells can modify the entire microenvironment of the lymph nodes by modulating the chemokine secretion and MDSC homing. And with that, I would like to acknowledge my mentor, Dr. Ethan Shevak, and my collaborator, Jeff Zhu, and all the lab members for discussions about this project, and Kay for sorting, and Zihan and team for helping me with sequencing and analysis. And with that, I am ready to take questions.
Thank you for your presentation, Doctor. It was really like very neat and clear explanation, and yeah, it was really good listening to it. So, do we have any questions for Dr. Shalini? I guess there are no questions right now, so it's okay. We'll just share your contact to everyone. So if they have any queries, so yeah, they'll definitely send you an email to
Sure.
the same, so you can just get back to them.
Sure.
So
Thank you.
yeah, with this, I guess we come to an end to our day one sessions, as we have two pending presentations where both of them are not available right now as we consider the time. It's really in the midnight for them, so we couldn't contact them. So we'll just take that to tomorrow, and we'll see you all tomorrow at the same time at uh, 10 a.m. in the morning at Eastern Daylight Time. Thank you one and all for your presence today. Have a great day ahead.